Hi everyone, am I audible, visible? Hello. Am I audible and visible to you? Good evening everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today it is the digestion and absorption part 2, and in detail we are going to discuss about the different parts of the alimentary canal, about the digestive glands, and then we will talk about the physiology of the digestion, right? So today's lecture is again an important lecture, and yesterday we have covered the basic part, like what is mouth, what is there in the buccal cavity, about the dentition, that's all. Okay, so today we will start from the pharynx, then we will talk about the esophagus, stomach, and in detail we are going to discuss about this particular chapter. So I hope you guys have revised the last class, and moreover I really don't know like what's your plan, students, because I can see very less number of students are there, right? And even you know that this chapter, it is human physiology, one of the one of the most important units. So please be regular, be consistent, start attending the lectures properly, and if a teacher is asking something like this question you need to answer in the comment section, please be sincere and do that. Okay, so I was just checking the comment section. Very good, Sinivasan, right? Thank you so much. Thank you so much for this, bache. You will get the same lecture. And Priyanka, as usual, wonderful, bache. You are giving the answer of every question. So I was asking about the foramen cecum. So see, foramen cecum of the tongue is a depression at the posterior part of tongue, right, bache? And it is the first and it gives rise to very important structure, that is your first endocrine gland. So it's a correct answer. Very good, and thank you so much, bache. Akanksha. So guys, be sincere, and please in the comment section, if any teacher is asking a question, do answer that in the comment section. It will not take you much time, trust me. And moreover, let me tell you this thing, you know, although you know that, bache, your HSP sir's batch, this batch, right, this batch is at a price of 1999, chemistry mind maps by HSP sir, and if you will join this batch, surely you guys are going to get 180 out of 180 marks in chemistry, let me tell you. And moreover, from tomorrow its price, right, its price will be near about 4,000 or 5,000, but today you are getting it just at a price of 1999. So please kindly, it's a request, students, right, it's a request, please be the part of this batch as soon as possible. Otherwise, you know, you all then request that, ma'am, again please decrease the price and all, then it will not be possible. So be the part of this batch, and trust me, if you will join this batch, you will score full house in the chemistry, right? Literally you are going to score that. Clear? Okay, and even I hope you have checked that. So do check this video. I have explained everything related to anti-NEET bill, that what exactly is this anti-NEET bill, should you is there any concern, right? You will get every possible detail of that anti-NEET bill in this particular video, so do check it out. Clear, bache? And especially if you are from Tamil Nadu, you should check it. Okay, so now let's start the lecture again. So digestion and absorption it is. So in the last class we have already discussed what exactly is the digestion, right? So let's revise it very quickly. So when it is the digestion, you know that we are breaking down the complex substance into the simplest one. That is what digestion is, right? That is what digestion is, that we are breaking complex substance, complex macromolecules into the simpler one, into the absorbable ones, that the form that can be absorbed by the cells. That is what digestion is, right? You know that in our diet, right? You know that in our diet, what is there? We used to eat carbohydrates, protein, fats. They are important for the energy, isn't it? And when you consider these carbohydrate, proteins and fats, they are macronutrients, right? We need them in more quantity. Okay, and if I talk about the carbs and the proteins, you know that they are macromolecule. So the cells of our body cannot absorb them directly when they are in their, you know, polymeric form. So what we have to do, body is doing what body will do, body will break it into simpler particles. Are you getting it? Body will break it into simpler particles, into simple forms, so that it can be absorbed by the cells of the body, right? That is what digestion is. Clear, bache? So now you know that when it is the digestive system, Yes. So when it is the digestive system, so digestive system includes what? It includes your alimentary canal and the digestive glands. Okay, and in the case of humans, right, we belong to class Mammalia. So in our case, advanced type of systems are there. So we have complete digestive system. Two different openings are there. There will be the mouth, there will be an anus, right? There will be a mouth, there will be an anus. Clear, bache? Clear, bache? And when it comes to the alimentary canal, you know how are we going to start? The mouth will be there. Okay, then buccal cavity. Fine. The pharynx. Esophagus, stomach, small intestine, then large intestine, and finally anus will be there. Right, finally there will be the anus. Clear, bache? Clear. And when you talk about the digestive gland, we will talk about the salivary gland, then your gastric glands, pancreas, liver, intestinal gland. Right, that's what we need to study. Is it clear? That's what we need to study, right, bache? So in the last class, we have already covered the mouth, buccal cavity, and in buccal cavity, the roof is palate, right? The floor of buccal cavity will contain tongue, and in buccal cavity, their teeth are also present, right? So can you just tell me the type of dentition in the case of human beings? Yes, can you just tell me the type of dentition in the case of human beings? The type of dentition in human beings, be quick. Yes, everyone, be quick. The type of dentition in human beings. Quickly answer it. Very good, Shri Harsha bache. So when it comes to the dentition, the thecodont, diphyodont and the heterodont type of dentition is there. Right? Thecodont, diphyodont and the heterodont type of dentition is there. Right, bache? And in the last class, that's what we have covered. So let's start with the pharynx. Fine. Let's start with what? Let's start with the pharynx. So now when it comes to the pharynx, even in the last class, I told you about it. We were drawing one diagram, remember? Do you remember this diagram? Yes, in the last class we were talking about it. So, bache, when you talk about the pharynx, you know that, right? Pharynx is a common passage. It is a common passage for the air as well. It is a common passage for the food as well. Okay, it is a common passage. It is a common passage for the air as well. It is a common passage for the food as well, right? Bache, for air as well as for the food, it is a common passage. First thing. Now you know that air will enter from internal nares, isn't it? External nares. External nares means these nostrils. Air is going to enter from this part, right, bache? So here, when it comes to the pharynx, we have three parts, and the trick for it is knoll. What is the trick? The trick for it is knoll. So N stands for nasopharynx. O stands for oropharynx. L stands for laryngopharynx. How many parts are there? Three parts are there. So we have nasopharynx, oropharynx, laryngopharynx. Nasopharynx, oropharynx, laryngopharynx. So now this nasopharynx, like see this external nares, external nostril, it will lead into this nasal chamber, into this nasal cavity, right, bache? So here, right inside you will find the internal nares. Internal openings are also there. Then it will lead into the nasopharynx, right? It will lead into the nasopharynx. And do you know that one thing, that in nasopharynx we have one more opening. Do you know that there will be the opening of eustachian tube? Are you aware of it? There will be the opening of eustachian tube. Do you know about it? Yes or no? Do you know about it? What are these eustachian tubes? Bache, like when we will start the chapter, soon we'll start the chapter neural control and coordination. We will discuss the structure of ear there. So when you talk about the ear, the middle ear especially, so there is going to be a tympanic cavity, and then you will see a tube-like structure, right? That eustachian tube. It is actually linking this ear to this pharyngeal part, right? To this pharyngeal part. So this eustachian tube will open here in the nasopharynx, and it is basically maintaining the pressure, right? So in detail we will talk about it in the structure of ear. Clear? So next to your nasopharynx is what? Next to your nasopharynx is your oropharynx. So in the last class also I told you about this, that oropharynx is a common passage for both, right? Oropharynx, it is a common passage for both. Common passage for the food as well, common passage for the air as well. So this buccal cavity will basically going to lead into this oropharynx, right? So see, from nasopharynx side it is receiving the air, from buccal cavity side it is receiving the food. Okay, so next to this oropharynx is the laryngopharynx. That's what I said, right? Next to it is what? It is the laryngopharynx. But before that you need to understand something else. Now, like this part is our dorsal side, this part is our ventral side, and this part is our lateral side. That's what we have. Dorsal side, ventral side, lateral side. Dorsal side, ventral side, lateral side. That's what we have. Yes or no? That's what we have. So now towards the ventral side we will see the trachea first, and then, like I'm talking about these tubes basically. Obviously the sternum is there, right? All that ribs are there, but after that what is present? The trachea, and just behind the trachea there is the presence of the esophagus. So if I talk about this diagram, right, if I talk about this particular diagram, because this is the mouth region, so here you guys can see this is the ventral side, and this side here it is going to be the dorsal side, right? This part is the ventral side, and this part here will be the dorsal side. Now what is going to happen? This laryngopharynx, right? This laryngopharynx, okay, it will further lead. Like let's say this is the ventral side, here it will lead into this larynx, okay, and just behind this larynx, this part here, it will lead into esophagus. It will lead into esophagus. So ultimately I am saying that your laryngopharynx, it will lead into that. From this laryngopharynx there are two tubes, right? How many tubes are there? There are two tubes. One tube is this, basically there will be the two tubes. So laryngopharynx, it will leads here into the larynx, here just behind the larynx, right? Just behind the larynx, right? Or you can say the dorsal to the larynx, it will lead into the esophagus, okay? It will lead into the esophagus. So here what do you need to remember? That the opening of this larynx, it is glottis. Opening of this larynx, it is glottis. What is it? It is glottis. And bache, the opening of this esophagus is what? It is gullet. Opening of esophagus is gullet. Opening of your larynx is what? It is the glottis. So glottis, gullet. Glottis, gullet. This is what you need to remember. So this laryngopharynx here, it is leading into the larynx. I'm repeating this again. Here it is leading into the esophagus, right? So esophagus is placed just behind the trachea. So here you can see the ventrally larynx, and then trachea is there, and just behind it, or just dorsal to this trachea, you will see the esophagus. So any doubt? Any doubt? So now there is one more question, that like when we swallow the food, right? When there is the deglutition, right? Then what is going to happen? This food can even enter in our trachea also, isn't it? This food can even enter in our trachea also, isn't it? So what is going to happen when we swallow the food? Bache, this glottis is opening, but it is covered with a cartilaginous flap, that is epiglottis, right? There is the presence of a cartilaginous flap, that is epiglottis. Let me show you. Like let's say, right? So opening of larynx is glottis, and when we swallow the food, Okay, that opening will be covered with a cartilaginous flap. Okay, and that is the epiglottis. What is it? That is the epiglottis. So if you will ever get confused, what you need to remember, epi means above. Right? What is the meaning of epi? Epi means above, above glottis. So it is something which is going to cover the glottis when we swallow the food. So any doubt here? Any doubt here? So this part, as I said, will be your trachea. Just behind your trachea, you will find another opening. That is your esophagus. So now we are going to talk about the esophagus. Done? Clear? Clear, clear, clear? What are we going to discuss? We are going to talk about the esophagus now. So what do you know about the esophagus? Yes. What do you know about the esophagus? It's a thin tube-like structure, isn't it? So this pharynx will leads into the esophagus, and this esophagus is a thin tube-like structure. And bache, in the esophagus, we are not going to find any particular glands which are meant for the which are secreting substances for digestion. It is not like that. In the esophagus, there will be the mucus gland only. What will be there? There will be the mucus gland only, right, bache? And with the help of peristalsis. What is peristalsis? Rhythmic contraction and relaxation of the smooth muscles, right? Rhythmic contraction and relaxation of this esophagus. Why is it possible? With the help of smooth muscles, it is going to pass the food downward, right? So that's what esophagus is a thin tube-like structure. If I talk about the length of the esophagus, it varies. It depends upon the neck region. Do you know that? It depends upon the neck region. Just in the case of giraffe, the esophagus is a very long tube. Why? Because the neck there is long. Are you getting it? Neck there is long. So that's what you need to keep in your mind, right? So before telling you about the esophagus, right, we should discuss one more thing, and that's histology of alimentary canal, right? According to me, if you know the histology, if you know about the layers, then you can easily understand this particular part. Okay, so esophagus, as I said, thin tube, right, near about 20-25 centimeter long in our case. So it will carry the food to the stomach. In the esophagus, only mucus glands are there, and there will be the peristalsis, that is rhythmic contraction and relaxation of smooth muscles, and the food will pass downward. That's what we know. But now here, before discussing about this esophagus, we should know about the histology. What we need to know? We need to know about the histology. Histology, you know that histology means study of tissue. Clear, bache? What is the meaning of histology? It means study of tissue. And when you talk about the histology here we are going to talk about all the tissues which are making our alimentary canal. Okay, so alimentary canal is basically a tube having esophagus, then stomach, which is the widest part of alimentary canal, then small intestine, large intestine is there. Clear, bache? So study of tissue is your histology, and here in alimentary canal you will see there are 4 layers. How many layers are there, bache? There are 4 layers, right? There are 4 layers. So there is a trick for these 4 layers, right? I hope you all know about SMS. Do you know about SMS? Yes. And it is a question, it's a repeated question. Do you know about SMS, short messaging services? These days we are using it for the OTP, bank OTP, for the delivery, for order delivery. The OTPs that we receive, it is generally via these messages and all, right? Right. So we use the term SMS for it, short messaging services. So instead of SMS, you have to keep it as SMS. What do you have to write? You have to write SMS. What do you have to write, bache? You have to write SM. So S here stands for serosa. M here stands for muscularis. Okay. S here stands for submucosa. And M stands for mucosa. These are the layers of your alimentary canal. Right, what do we have? We have SM, serosa, muscularis, submucosa, mucosa, serosa, muscularis, submucosa, mucosa. I'm repeating this again. Serosa, muscularis, submucosa, mucosa. These are the 4, right? These are the 4 layers that we have. And these 4 layers, bache, in different part of the alimentary canal, they are modified as per the need. Right, like if I have to give you one example, in the case of small intestine, you will see that mucosa will form finger-like enfoldings, and we are going to call it as villi. In stomach, you will see that this mucosa will form some extensions, some no doubt they are also finger-like, and we used to call it as rugae. So that modifications of layers, they are different in different parts. Okay, that modification of layers, they are different in different parts, but that's how it is present. Clear? That's how it is present. So it's SM, serosa, muscularis, submucosa, mucosa. Serosa, muscularis, submucosa, mucosa. Are you getting it? So these are the 4 layers, bache, right? So in the NEET examination, they can ask you the sequence of these layers. So outer to inner side, this is what you guys are going to find. So outermost is serosa, then muscularis, then submucosa and mucosa. Now this serosa is also known as visceral peritoneum, right? This serosa is also known as visceral peritoneum. What is it? It is visceral peritoneum. Now the question here is, what is peritoneum? I am assuming that you know about the peritoneum. Do you know about the peritoneum? Peritoneum is the outermost layer. What is peritoneum? Outermost layer, which is covering the body organs, right? Obviously its main function is to provide the protection, and this visceral peritoneum, it is mesothelium. Now if you have attended the class of structural organization in animal, then you people know, right? Mesothelium means the epithelial tissue is made up of mesoderm. The epithelial tissue is made up of mesoderm. So these are the layers which are present: serosa, muscularis, submucosa and mucosa. So serosa is the visceral peritoneum. But let me tell you, in esophagus, okay, in esophagus, this serosa is known as, this is extra bache, is known as tunica adventitia. Do you know that? Here it is known as tunica adventitia. What is it? Here it is known as tunica adventitia. Is that clear? Is that clear? Actually, bache, because in the case of your esophagus, some part of this esophagus, some layers here, right, the initial portion, they are voluntary. It is not involuntary, it is voluntary. Only the initial part, right? Right, only the initial part. Are you getting it? Right, only the initial part. It is voluntary, rest is involuntary. So here in the case of esophagus, you cannot properly differentiate the serosa and muscularis, right? You cannot differentiate these layers properly, it is believed. So that is why we are using the word tunica adventitia here. Clear, bache? Clear, bache? We are using the word tunica adventitia here. Done? Done? And actually the epithelium is multi-layered here. Fine, it is multi-layered here, that's why. That's why. So it is a tunica adventitia word. Now let's understand this part first, okay? Let me show you the diagrams. So see this. So this is what we are discussing as of now. Histology. Okay, histology. That's what we are discussing here. So here you people can see, right, here you people can see the outermost layer, and then these layers are present. And inside you know that there is the lumen. What is there? There is the lumen. So lumen is that empty space. Okay, lumen is that empty space. So that is covered by, as I said, kids, I'm repeating it again, mucosa, then submucosa, then muscularis, and then visceral peritoneum. Fine, visceral peritoneum. Okay, visceral peritoneum. So visceral peritoneum, also known as serosa, your muscularis, your submucosa, innermost is your mucosa, and then comes the lumen here. Is it clear? Yes, is it clear? This is important. Yes, it is important. Just look at it. Look at the arrangement here. Right, just have a look, bache. Can you see these layers? Yes, outermost, then. So these layers are having blood supply. These layers are having your nerve supply as well. Muscles are also present, right? What do we have here? Even muscles are also present. So now firstly, let's talk about it, right? Then I will show you the diagram. So serosa is very simple. Okay, the serosa part is very simple. And what is that? that it is the outermost layer meant for the protection? Yes or no? When it is the serosa, it is the outermost layer, right, bache? And it is meant for protection, yes or no? It is meant for protection, yes or no? Done? Done, bache? Outermost layer, and it is made up of mesothelium. Okay, right? And yes, some connective tissue is there. Next to it is your muscularis. Now, bache, muscularis means muscles, isn't it? Muscularis means muscles, isn't it? Yes, isn't it? Muscularis, it means muscles. Yes or no? Yes or no? So here in the alimentary canal, the trick is ICOL. ICOL. So muscles are present. Inner circular, right? It's ICOL. The trick is ICOL. Inner circular, outer longitudinal. Inner circular, outer longitudinal. Clear, bache? I'm repeating this again. Inner circular, outer longitudinal it is. Clear? Clear? So these are the muscles which are present here in the alimentary canal, and these are the smooth muscles. What type of muscles are there? What type of muscles are there? Smooth muscles. So I hope you know that these smooth muscles, they are fusiform, they are uninucleated, spindle shaped basically, and these muscles are involuntary. You cannot control their function with your own will. Fine. You cannot control their function with your own will, right? They are under the control of the nervous system. So what do you have? Inner is the circular muscle, outer is the longitudinal muscle. So these smooth muscles, right? Obviously, digestive system is having its own nerve plexus, right? It is having the network of nerve fibers as well. Basically, it is the enteric nervous system which is innervating our digestive system. Okay, which is innervating our digestive system. Clear, bache? So that nerve fibers are going to help here in the contraction, in the rhythmic. What is the word? Rhythmic contraction and relaxation of the Anuradha, bache, long time in the rhythmic contraction and relaxation of the smooth muscles. Contraction, relaxation, and that is what peristalsis is. Clear? That is what peristalsis is. Like here in the case of esophagus, that's what I'm telling you, that esophagus is going to pass the food downward. है ना? It is going to pass the food downward. It's a tube-like structure, and this tube-like structure will open up here in this J-shaped stomach, right? So here you will see that there will be the peristalsis. So peristalsis is what? Rhythmic contraction and relaxation of the smooth muscles, and because of that, food will pass downward. Clear? Right? Food will pass downwards. Clear, bache? Sure? Is it clear? Yes or no? Done? So ICOL, this is what you need to remember. Moreover, when you talk about the stomach, in stomach, this is important, please note down. In stomach, in addition to inner circular, outer longitudinal, in addition to, in stomach, please note down, in stomach, in addition to inner circular, outer longitudinal, yes, inner circular, outer longitudinal muscles, oblique muscles are also there. Fine. Oblique muscles are also present. And in stomach, bache, the muscles, they are quite thick. Why? For the churning of food. Clear? For the churning of food. Churning of food means mechanical digestion of food. Clear? So in addition to inner circular and outer longitudinal muscle, oblique muscles are also present. Where? In stomach. Right? Like here, outer longitudinal, inner circular, then inner to it is oblique muscle. Fine. Oblique muscles, they are present. Clear, bache? Clear, bache? So this is about the muscularis. Clear? This is about the muscularis. Muscles are present there. Fine, muscles are present there. Now, bache, it is not just the muscles that are present here in muscularis. In muscularis, network of sympathetic and parasympathetic nerve fiber is also there. The network of sympathetic and parasympathetic. Do you remember this? If yes, then good. Otherwise, don't worry. In human physiology, in chapter neural control and coordination, we will discuss about it. So, as I said, in muscularis, network of sympathetic and parasympathetic nerve fibers is also present. Fine. It is also present. It is present in between that longitudinal and circular muscles, and in stomach in between that circular and oblique muscles. So this network is present. This network, nerve fibers, you know that they are important, isn't it? They are important. Clear, bache? That nerve fibers are going to tell that muscle that now it's the time for the contraction and the relaxation. So here, this network of nerve fibers, it is known as, yes, bache, it is known as myoenteric nerve plexus. Plexus means network, or back plexus, or a back plexus. These are the two words. Myoenteric nerve plexus or a back plexus. What do we have here? Myoenteric nerve. Yes, bache. What do we have here? Myoenteric. Quick. Myoenteric or a back nerve plexus. It is important. Please write down. Done? Yeah, so yes, in muscles, in muscularis, this is what we have. Clear, bache? In layer muscularis, this is what we have, and it is present in between the longitudinal and circular muscle. Even in stomach, it is also present in between the longitudinal and the circular muscle. That's what you need to remember. So it is a network of nerve fiber. So obviously it will help in peristalsis. It will help in the rhythmic contraction and relaxation of what, Sheetal? It will help in the rhythmic contraction and relaxation of the smooth muscles. Medlike, bache, oblique muscles, like we have circular, we have longitudinal muscles, same way we have oblique muscles as well. And that type of muscles are there in your stomach only. Where will you find it? In the stomach only. So this part is important. Please write down. Myoenteric or a back. Right? So if you will ever get confused, just remember myo word is associated with muscle. Okay? Myo word is associated with muscle. So it will be there in the muscularis layer. Simple as that. So digestive system is having its own enteric nervous system. Right? Which is going to take care of all that secretions and everything. Done, bache? Done, bache? So that's what you need to know. Clear? That's what you need to know. So next to it is submucosa. Next to it is submucosa. Below mucosa it is. Fine. Below mucosa it is. It is submucosa. Now here in this layer, bache, what will you get? Obviously the connective tissue. Yes, in the submucosa, what are you going to get? The connective tissue. Right? You will get the blood vessel, lymph vessel, nerve fibers here, and you will even get areolar connective tissue. Areolar, you know that the connective tissue having air spaces, right? The connective tissue having air spaces. So areolar connective tissue, lymph vessels, nerve fibers, okay, blood vessels are present, isn't it? All of them are present here in this submucosa. Is it clear? All of them are present in submucosa. And when, bache, you talk about the nerve fibers, so I told you, no, there will be the network. So here again, there will be the network of nerve fibers, and that network, for this network, the word here is Meissner plexus. Meissner's plexus. Clear, bache? So it's a network of sympathetic and parasympathetic nerve fibers. Are you getting it? It is a network of sympathetic and parasympathetic nerve fibers. Clear, bache? So these are Meissner's nerve plexus. right? These are the Meissner's nerve plexus. It is the submucosa. So submucosa is having what? It is having the connective tissue, right? Submucosa is having what? It is having the connective tissue. Any doubt here? Any doubt here? So network of nerve fiber, Meissner plexus is there. So areolar, connective tissue, lymph vessel, nerve fiber, blood vessels, all of them are present. Clear, bache? All of them are present here. Are you getting it? Sure? Sure, everyone? Sure. So one point that you all should note down. See, next to submucosa is mucosa. Next to submucosa is mucosa. This mucosa is itself divided into 3 layers. Like lamina propria is there, connective tissues are present, right? Right. But we are not going to discuss that in that detail. Okay. So even in the mucosa, we even see that muscles are present in mucosa layer. Clear, bache? Lamina propria is there in the muscle layer, in this mucosa layer, right? Modified lymphatic tissues are also there in this mucosa layer. Clear, bache? So there are many layers in this mucosa. Matlab, it is itself divided into 3 parts, right? It is itself divided into 3 parts. But as I said, we are not going to discuss that in depth, right? But what we need to focus, see, let me tell you. This is mucosa, next to it is this is submucosa, next to it is mucosa. Mucosa is having the glands, okay? Like your mucus gland is there. In stomach, we will talk about the gastrin gland, okay? We will talk about the Pennant cells. So such secretory glands, such secretory cells, they are present here in this mucosa. Are you getting it? They are present here in this mucosa. So obviously, if we want them to secrete their, to pass on their secretion, so this submucosa, like submucosa is having these network, right? Submucosa is having this network of nerve fiber. So somewhere it is helping for passing out the secretion from the mucosal glands as well. Okay? Okay? So here in muscularis, this network of nerve fiber is helping in the peristalsis. Here, this network of nerve fiber is somewhere helping in the release of the digestive juices. In release of the digestive juices. Are you getting it? Are you getting it? So just note down one thing. I was about to tell you in the stomach, in the small intestine part, but in your notes you can include here, right? So as I said, all glands are there in mucosa, but we have one exception. You know, Brunner's gland of duodenum, right? Brunner's gland of duodenum. You know that duodenum is the part of small intestine, isn't it? Duodenum is the part of small intestine. So Brunner's gland of duodenum is present in submucosa. Okay, so it is a kind of exception. Fine. It's a kind of what? It's a kind of exception. Clear, bache? Clear, bache? Yes. Yes. So Brunner's gland of duodenum, which is the part of small intestine, is present in the submucosa layer. Okay. And this Brunner glands, it will release, right, alkaline secretions. They are non-enzymatic secretion. So obviously, it is also making the secretion of the intestinal juices. So that's what we need to remember. Any doubt? Yes, that's what we need to remember. So this is about the submucosa. And again, as I said, this part is important, right? MCQ can come from this part. Done. MCQ can come from this part. So Brunner's gland of duodenum, which is the part of small intestine, is present in submucosa. Rest all the glands are there in mucosa. Clear, bache? Clear? Sure? Sure? So now when it comes to the mucosa, as I said, even this mucosa is divided into 3 parts. Like we have the outer part here, middle part here, and the inner part here. Clear? Clear? So middle part is basically your lamina propria. And it is nothing here. You will find the connective tissues. Outer layer will be, it is meant for the protection, of course. Right? What will be the role of this outer layer? It is again for the protection. And this outer layer is also having, yes, bache? Done? This outer layer is also having, it is also supporting the folds of alimentary canal. It is having the muscles. This mucosa layer is itself containing some muscles. Okay, so muscles are also present in mucosa. Clear, bache? Muscles are also present in mucosa. So middle layer is lamina propria. So here you will see the connective tissue. Innermost layer where you are going to find the glands. Fine. Where you are going to find the glands. So it is actually forming the lining, the inner lining of that alimentary canal. Are you getting it? It is going to form the inner lining of that alimentary canal. Clear, bache? Clear, bache? So whenever you will talk about the mucosa, as I said, even in mucosa, the inner layer is having the glands, the secretory. So let me tell you, bache, here the cells are going to be columnar. What type of cells will be there? Columnar epithelium will be there. Columnar cell means tall and cylinder, and nucleus is present at their base. Right? What is the meaning of columnar cells? Tall and cylinder, and the nucleus is present at their base, bache. Right? So it is the innermost layer. What is it? It is the innermost layer. So this layer, always remember, right? This inner layer except esophagus, because in esophagus it is multicellular. Except esophagus, it is it is single layered, uniserrated, or single layered, whatever you want to write. Clear, bache? Clear? So you will see even in the stomach, even in the small intestine, we will even see the in small intestine the brush bordered epithelium, but that will be of columnar cells. Right? So that's what we are going to see. Right? So this part clear, bache, now? So just look at the diagram. Look at the diagram. Outermost serosa, inner circular, outer longitudinal. Right? Submucosa, mucosa, then lumen. So here in NCERT you can mention Auerbach plexus, also known as myoenteric plexus, network of parasympathetic and sympathetic, and here you will find Meissner's plexus. What will you find here? You will find Meissner's plexus. Clear? You're going to find Meissner's plexus. Okay, done? Any doubt? Any doubt? So your stomach is containing ICOL, no doubt, but oblique muscles also. Done? But the oblique muscles also. Fine, bache? But the oblique muscles also. Clear? Clear? So now come back to this portion. Now you will understand the layers in a better way. So what's that? That's esophagus. So I told you already about the esophagus. The thin tube it is, right? Peristalsis will be done. So now I hope you know how this peristalsis will be done. Okay? Okay. So now you know that you will eat something. You are going to chew it. The saliva will be mixed in the buccal cavity, and then that food will pass to the stomach. So actually it is the bolus, right? When in the buccal cavity you masticate the food and the food get mixed with the saliva, then what is that food? That is the bolus. What is it? That is the bolus. So it is the bolus which will pass down. Clear? It is the bolus that will pass down. Clear, bache? Clear, bache? So as I said, the length of the esophagus depends upon the neck region. So in the case of giraffe, it's very long. Okay. In the case of giraffe, it's very long. Okay. So this tube. Now you know that esophagus. Now you know that the esophagus will leads into the, right? The esophagus, it will lead into the stomach, isn't it? It will lead into the stomach. So actually, bache, in our body, if you know it very good, otherwise let me tell you. They say here you have the thoracic cavity, where lungs, heart, even the esophagus is also there. Then comes the abdominal cavity. Then comes the abdominal cavity, and you know that in between these two cavities we have the diaphragm, isn't it? What do we have? It's a unique feature of mammals. It's a unique feature of mammals. Diaphragm is there. It's a muscular tissue. Diaphragm is there. Remember, it is separating your thoracic cavity to your abdominal cavity. Okay, it is separating the thoracic cavity from abdominal cavity. So this esophagus from this part, right? In the diaphragm there is an opening, right? That esophagus will cross that opening and finally open up here in the stomach. So that opening here is known as hiatus. Not important, you can miss this part. It is just for your knowledge. Clear? This opening here in the diaphragm is hiatus, by which your esophagus will go down. It will open up there in the stomach. Is that clear? Is that clear? Yes. Now, first of all, Sahil Mumin, I think, right, as per my knowledge, the biology is not the part of IIT exam, isn't it? You already have maths, physics, and then chemistry. Why do you want the biology there? Why do you want the biology there? मैम आप बहुत अच्छा पढ़ाती हो. शुक्रिया साहिल, बहुत खुशी हुई आपके अप्रिसिएशन से. बहुत मजा आया, बहुत ज़्यादा अच्छा लगा. Thank you. Anything else? And Sheetal बच्चे, you have to make your notes. Okay, although I will share it in my personal Telegram group also, that is Love Biology by Ambika, and even in our official Telegram group Unacademy NEET English, you guys can join. I'll share the PDF there. Fine. But with this lecture, what you have to do? You have to make the notes, running notes. Fine. Okay. So anything else? Any doubt? Okay. You will get the DPP too, Sheetal. So thoracic cavity, abdominal cavity is there. Any doubt? Sure. So next to it is what? It is the stomach that we need to discuss, and stomach is the widest part of your alimentary canal. Stomach is the widest part of your alimentary canal. Clear, bache? It's a J-shaped pouch-like. Structure, and it is what? It is the widest part of alimentary canal. Clear, bache? Clear, bache? So stomach is having 4 parts. One is your cardiac part, then fundus, body, and the pyloric part or pylorus. Clear? These are the 4 parts of the stomach. Cardiac part is there, fundus is there, body, pyloric part or pyloric stomach is there. Clear? Let me show you this. So now you know that esophagus, bache, it will open up in cardiac part. Where will this esophagus open? It will open up in this cardiac part. This upper covered part is nothing, it is the fundus, right? This part here is the body, and then this part here, this narrow portion here is the pyloric stomach or pylorus. Clear, bache? Pyloric stomach or pylorus. So esophagus will open up here in the cardiac aperture. In the cardiac part. Okay. Esophagus opens in cardiac part. Clear? It opens in cardiac part. So here we have one aperture, and this aperture here is, it is bache, yes, do you know the name? It's very easy. It's your gastroesophageal aperture. Okay, what is it? It is your gastroesophageal aperture, and you can also call it as cardiac aperture. What is it? It is cardiac aperture. What is it? It is cardiac aperture. Clear? Esophageal aperture and the cardiac aperture, and yes, you can, right, you can also call it as orifices. Orifices means opening. Done. You can also use the word orifices. Orifices means opening, right? So cardiac aperture, gastroesophageal aperture, cardiac orifices, it's all same. So it is showing, right, it is a sphincter or it is an aperture present at the junction of this esophagus and the cardiac part. Clear, bache? Now in the babies, you know na, if you give them milk, they will give you curd. Huh? Do you know that? In the case of babies, right, that used to feed on mother's milk, you know that if you will give them milk, they will give you the curd back. है ना? And immediately they will give you the curd back. Why is it so? Because in their case, what is going to happen, bache? Obviously they will have that milk, the digestion of that milk will be there, and in their case this gastroesophageal aperture is very weak. Okay, it's not that strong. Okay, so that is why when after feeding the babies, we used to pick them up like this, so that they should not give that curd back to us, but again they will. Eventually they will give it back to us. Okay, why? Because in their case, this gastroesophageal sphincter, na, it's very weak, so it will regurgitate, right? That curd will regurgitate. Are you getting it? So next to it here is this pyloric sphincter or pyloric orifices. It is pyloric sphincter. You can also call it as pyloric orifices. So this is the opening, and from here the esophagus will lead into the small intestine, right? It is going to lead into the small intestine, and which part of small intestine? It's duodenum, right? Which part of small intestine? It's duodenum. So pyloric stomach will pass into the duodenum. Clear, bache? It will pass into the duodenum. Done? Done, done, done. So esophagus, the widest part, the J-shaped pouch-like structure having 4 parts, cardiac, fundus, body, and the pyloric part. Right? Right? So now here in the case of the stomach, this stomach is also covered with the peritoneum. Okay, its outermost layer. Now you know about the layers as well, isn't it? You all know about the layers as well, right? I don't think that I need to explain it again. That SM is there, and in muscularis, here, in addition to circular and longitudinal muscle, oblique muscles are also there in stomach. In addition to circular and longitudinal muscle, oblique muscles are also there in the stomach. And in the case of stomach, the muscles, they are quite thick. Why? For the churning of food, for the proper churning of food. And here in the stomach, yes, this mucosa is going to form some finger-like structures, right? Some ridges-like structure. We used to call it as rugae. Right, we used to call it as rugae. So what is the modification of mucosal layer here in stomach? What is the modification of mucosal layer here in stomach? In the stomach, what do we have? We have these rugae. So when your stomach is full, then rugae will not be there. If your stomach is empty, then these rugae are present. Clear, bache? Thank you so much, Yash. Right? Right, so these are the rugae. So now I'm saying that the outermost layer, the stomach, your stomach is covered with peritoneum. Okay, it is covered with peritoneum. You know na, serosa and visceral peritoneum is same. Hai na, serosa and visceral peritoneum is same. I hope you know that. So this peritoneum here, it is having the depositions, right? Your adipose tissue. Adipose tissue means we are talking about the fat. Right, adipose tissue, lymph vessels, right, all of them are also covering this peritoneum. Are you getting it? In the case of stomach, right, peritoneum is having your fat tissue, adipose tissue, the lymph tissue is also there. Right? Adipose tissue and lymph tissue is there. Clear, bache? This peritoneum is like this. So in the case of stomach, in stomach, such peritoneum is known as, yes, such peritoneum is known as omentum. What's the word for it? It is known as omentum, right? It is known as omentum. Is it clear? Is it clear that in stomach, such peritoneum is known as omentum? Because peritoneum is having fat tissue, it is having the lymph tissue, so that is why we are calling it as what? It is omentum. And remember, the stomach is present on the left side of this abdominal cavity. Okay, where is it present? On the left side of this abdominal cavity. Clear, bache? That's the stomach having 4 parts, and you know about the layers as well. Clear? And you know about the layers as well. So I think we should discuss the gastric glands here only. right? Even although we need to discuss the salivary glands yet, but ठीक है, now we are talking about the mucosa. Is that okay if I will tell you about the gastric glands here? Is it okay? Hanji, is it okay? Okay, so gastric glands. So now you know that innermost layer, your mucosa is having it, isn't it? Isn't it? Yes or no? Yes or no? So they are basically in mucosa. So you have mucosal gland, and they are unicellular, let me tell you. They are unicellular tubular glands. Do you know that these gastric glands are what? They are unicellular, right, bache? They are unicellular, and even some of them are unicellular. I won't say all, but yes, they are microscopic and they are tubular. Fine, they are microscopic and they are tubular. Clear, bache? Microscopic and they are tubular. So let's discuss the gastric glands here only. So mucus gland, you have parietal cell. These are main. Parietal cell, also known as oxyntic cell. Peptic cell is there, which is also known as zymogenic cell, and even G cells are also present. Basically, these G cells are enteroendocrine cells. They are going to release the hormones. What are they going to release? They are going to release the hormones. Clear, bache? So mucus cells are, yes, they are even known as the goblet cell. It's one and the same thing, right? You can write it like this. Mucus cells, they are also known as the goblet cells. They are meant for the release of the mucus, of course. So mucus or goblet cell, parietal or oxyntic cell, peptic or zymogenic cell, and G cells, basically these are the type of enteroendocrine cells. So it is not mandatory that in the case of stomach you will have only one type of enteroendocrine cell, right? Right? Arey, my bad. Look, bache, why are you crying? They are also known as chief cells. Fine, they are also known as chief cells. Now clear? So chief cells, zymogenic cells, and peptic cells. It's all same. It's all same. Okay, so mucus cell also known as goblet cell, parietal cell also known as oxyntic cell, chief cells also known as peptic cell, zymogenic cell, and G cells, enteroendocrine cells. So as I said, there are many enteroendocrine cells, right? It's not just only G cell. Other than G cell, even D cell, EC cells are also there. Okay, EC cells are also there, but that we will discuss later. Okay, so now these mucus cells, what will they secrete? They will secrete the mucus. So what is mucus? What is mucus? That's your homework. You will tell me about the composition of the mucus, right? No doubt we are again and again saying that even in the esophagus, the mucus glands are there, right? They will help in the lubrication of our food, which is true. Right, which is true, it is helping in the lubrication of our food. But my point is, what exactly is the mucus? What that mucus is containing? Yeah, it's a fluid. You are right. But what exactly is the composition? That is what you people will tell me in the comment section. That is going to be your homework. You have to type it in the comment section. So mucus glands are going to secrete the mucus, and that mucus protects the inner lining of the stomach. Right? What will it do? It will protect the inner lining of stomach. Are you getting it? Inner lining of stomach will be protected by it. Okay. Okay. Now, parietal cell and the oxyntic cell. These two are very important. Okay. These two are very, very, very important. So here, these parietal cell or oxyntic cell, they have different role. Number one, it secretes HCl, the most important function. It secretes HCl, hydrochloric acid. But whenever you talk about the stomach, see, stomach is also known as gaster, right? It's also known as gaster, and these gastric glands, they will make gastric secretions, means stomach secretions. What will they do? They will make gastric secretions, that is your stomach secretions, and gastric secretions are acidic. You can say the pH here is in between 1 to 2 or sometimes 1 to 3. Right? These secretions are what? These secretions are what? Acidic secretions. Stomach secretions are what? They are acidic secretions. Are you getting it? So pH is 1 to 3, or you can say that, let's keep it 2 to 3. 2 to 3. Right? Acidic pH is there. So why stomach secretions are acidic? Because of the presence of the HCl. So in the NEET examination, they can ask you this question. It is important. Right? It is important. They can ask you this question, that which cells are going to secrete the HCl, and that is your parietal cell, also known as oxyntic cell. Right? That are your parietal cell, also known as oxyntic cell. Clear, bache? Clear, bache? Yes or no? Yes or no? It secretes HCl, right, which is going to make the stomach secretion, what? Acidic. And you know, this HCl is playing different roles here, right? I still remember, I think it was a question in class 10. What is the role of HCl? This HCl is very important. It kills microorganisms present in bolus. Do you know that? This HCl, it kills microorganisms present in bolus. Okay, this HCl, do you know it inactivates? Please write down. It inactivates salivary amylase present in bolus. Do you know that it inactivates salivary amylase, which is also known as ptyalin? So in the stomach, there will be no digestion of the carbohydrate. Okay? Right? Right. Clear? And even this HCl helps in the activation of inactive proteases. Like in the stomach secretion, we have some protein digesting enzymes. So that will be activated by the HCl. So that is the role of HCl there, which is very important. Clear? So it will convert inactive enzymes into the active enzymes. Clear, bache? Clear, bache? So it kills microorganisms, inactivates salivary amylase, helps in activation of inactive proteases, and moreover, it softens the food, right? So provide a medium for what? For the digestion. Take it. So that's the first point. And now another main function, okay, another main function of these parietal cell and oxyntic cell is it releases Castle intrinsic gastric factor. Castle intrinsic gastric factor. Do you know that? Right? Do you know that? Chalo, let me use another slide. Parietal cell, also known as oxyntic cell. So as I said, bache, it secretes HCl. Now you know the role of HCl, isn't it? You know the role of HCl, right? And along with that, it also releases Castle intrinsic factor. Right? It will release what? It will release your Castle intrinsic factor. That is your CIF. And this Castle intrinsic factor helps in the absorption of vitamin B12. Clear, bache? It helps in the absorption of vitamin B12, which is a water soluble vitamin. Okay? Which is a water soluble vitamin. Let me explain this part. See, you know that we always used to say that curd is more nutritious than milk. I hope you know about it, right? That curd is more nutritious than milk. Just in India, there are many people who are intolerant to lactose. They cannot digest the milk, right? Milk contains lactose sugar. I hope you guys have attended my biomolecule session. So when you talk about the milk contains the lactose sugar, and in India many people are lactose intolerant. They cannot digest the milk, they cannot digest the lactose present in the milk. So in that case, it is the curd which is highly nutritious. And even if we compare, even if you are not the lactose intolerance, even if you will compare the milk and the curd is more nutrition. Curd contains vitamin B12. Okay, and how that vitamin B12 will be absorbed? It will be absorbed with the help of this Castle intrinsic factor. How can we absorb it? We can absorb it with the help of this Castle intrinsic factor. In short, you can also call it as CIF. Fine. So if the CIF is not there, our body cannot absorb the vitamin B12. And let me tell you, this vitamin B12 is very important for the formation of your RBC. Do you know that? It is very important for the formation of your red blood cells, right? Red blood cells. Even bache, it is a MCQ as well. Do you know that? It is a MCQ as well. Are you aware of it? Right? That which cell, the absence of which cell can result in anemia. So it is your, yes, it is your, this cell, this particular cell, right? Clear, bache? So it will result in anemia, and you have to tell me that which anemia will be caused by the absence of vitamin B12. Fine? There is a particular word for this particular anemia, and that's what you need to tell me. Okay, that which anemia will be caused by the deficiency of your vitamin B12. This you will talk about microcystic anemia, macrocystic anemia. So this is what you need to find out. No, it is not sickle cell anemia, let me tell you. Right? That's not sickle cell anemia for sure. That's macrocystic anemia. Done? Okay. Okay. Sure. So vitamin B12 is important for formation of RBC. No vitamin B12, it can result in a type of anemia. I have given you the hint, but rest you need to find out the answer. Okay, just allow me a minute. Done? So that's anemia. Done, bache? Okay. So this is about the parietal cell and also known as oxyntic cell. Okay. It is about the parietal cell, also known as oxyntic cell. So another important type of cells are your peptic cell, chief cell, also known as zymogen, zymogenic cell. First of all, bache, do you know the meaning of zymogen? Do you know the meaning of zymogen? What is the meaning of zymogen? Quick. Do you know the meaning of zymogen? Do you know the meaning of zymogen? Which zymogen means inactive form of an enzyme. What is it? It is an inactive form of an enzyme. Right? Your enzyme is not activated. It is inactivated. It is the inactive form of an enzyme. For that, we use the word zymogen. So now when you talk about the peptic cell, this peptic cell is going to release the enzymes. Which enzymes will be released by this peptic cell? This peptic cell will even release your gastric lipase. But that gastric lipase, now the word is gastric lipase, it will help in the digestion of your fats, isn't it? Gastric lipase, it will help in the digestion of your fat, isn't it? Yes or no? Isn't it? Right? But so you can say that it is helping in the fat digestion, but that role is kind of negligible. Right? That role is kind of negligible. Gastric lipase, you can say that it can just convert 1% fat, right? It can just convert to kind of 1% fat. So I can say that no doubt gastric lipase is helping in fat digestion, but that is kind of negligible, right? It is having a negligible role. Imagine now only 1%, and moreover, these peptic cells, they will also release some inactive enzymes, inactive proteases. Now you know the meaning of proteases, protein digesting enzymes. So usually, bache, protein digesting enzymes, they are not present in the active form. What is the reason behind? The reason is if they will be in the active form, they can even destroy the framework of the tissue, isn't it? Ultimately, in the extracellular matrix also, you know that protein is there. So protein, right, it is playing a role in the framework. Okay, so these proteases, they are in the inactive form. They will only be activated when required. Okay, they will only be activated when required. So these are protein digesting enzymes. So we have two names here. One is your prorenin. It is R-E-N-N-I-N. Another is your pepsinogen. Okay, it is R-E-N-N-I-N. Remember, renin, R-E-N-N-I-N. Single, we have one more renin, R-E-N-I-N, right, which we will discuss in the case of kidney. Here it is R-E-N-N-I-N. Okay, so it is an enzyme, right? So inactive proteases, peptic cell is going to form these inactive proteases, prorenin and pepsinogen. So in the presence of HCl, that we will discuss later, it will form renin. Okay, and this pepsinogen will form pepsin. These are their active forms. Clear, bache? These are their active forms. So that we will discuss in the physiology of the digestion. Clear, bache? That we will discuss in the physiology of the digestion. So ultimately, they are going to help in the digestion of proteins, and this renin specially, it will help in the digestion of milk protein. Clear? Particularly, it will help in the digestion of milk protein casein. Done? We will discuss it in the physiology of the digestion. Fine? Fine. So that's what you need to remember. So now you know the role of your parietal cells as well. You know the role of your oxyntic cells as well. Fine? You know the role of these. That's important. Fine? That's important. Okay? Sure? Sure. Right? So, G cells, enteroendocrine cells. So see, as the name is suggesting, enteroendocrine gland. So it will release the hormones. So this G cells, they will release the gastrin hormone. Which hormone will be released by G cells? It's the gastrin hormone. What is it? It is the gastrin hormone. Clear, bache? It is the gastrin hormone, right? So this gastrin, it induces gastric secretions. Fine, it induces gastric secretions. Bache, I will tell you about the renin, their presence in infants and adult, everything, but that part will be discussed in the physiology of the digestion. Okay, Mr. Leisure Bunny. Okay, that will be discussed in the physiology of the digestion. Clear, bache? Clear, bache? So it is not just G cells which are present in the stomach. No, right? It is not just the G cells which are present in the stomach. In the stomach, you also see D cells. They will release somatostatin, inhibitory. Okay, they also have, you know, some another cells also. But mainly, we discuss about G cells. Why? Because this is the hormone which induces the gastric secretions. Okay, it is the hormone which induces, which stimulates the secretion of gastric secretions or secretion of digestive gastric juices. Clear? So the function of mucus, parietal, chief cell and G cell can be asked in paper. So it's important. Right? Ma'am, I'm Priya. Please tell my name. Wow. Priya. Now you're happy? Now you're happy? Other students should also start. Ma'am, my name is Bala. You should take my name. Ma'am, my name is Leisure Bunny. You should take my name. Wow. Our students are so mature. Wow. Too good, too good. Priya is saying, I'm Priya. Please tell my name. Priya, you are not happy to take your name. and you are asking me to take your name. वाह, बहुत बढ़िया. चलिए, so next. So that's all about the stomach, the gastric secretions. Okay, the next is small intestine. Next is small intestine. So now you know that pyloric part of the stomach will leads into the small intestine. So for small intestine, what do you need to remember? It's D, J, I. What is there in the small intestine? It's D, J, I. It's D, J, I. Right? D, J, I it is. Small intestine. Okay. So D stands for duodenum. D stands for duodenum. J stands for jejunum. I stands for ileum. Okay. It is I-L-E-U-M. We have one more ileum. That's different. That we will discuss in girdles. So here it is DJI. Okay. Small intestine is DJI. Duodenum, jejunum, ileum. Duodenum, jejunum, ileum. So your pyloric stomach will leads into this duodenum part. Right? It will lead into this duodenal part. Clear, bache? Clear, bache? So this duodenum is C-shaped. Actually, bache, it is U-shaped also, but we have to follow the NCERT. So we will write it as C-shaped. Clear, clear, clear? It is C-shaped. Done? Okay. Fine. So duodenum, it is the proximal part of the small intestine. It is C-shaped. It is also U-shaped, but in NCERT, it is written as C-shaped. So we have to follow that only. Clear? Then comes the jejunum, middle coiled part. Right? It is the middle coiled part. And then comes the ileum. So ileum is highly coiled. Right? It is highly coiled part. Okay. So yes, that's what you guys can write down as well. Just a minute. So duodenum. First word is what? It is a duodenum. So as I said, C-shaped, less vascular. The blood supply is not that abundant here. So minimum absorption is there in duodenum. Fine. Minimum absorption is there in duodenum. Then comes your jejunum and ileum. So now you know that coiled part, and this one is highly coiled. This one is highly coiled. So students, can you tell me where will be the maximum absorption? Yes, where will be the maximum absorption? Where will be the maximum absorption? This coiled part. It is more vascular because here we have abundant blood supply. So obviously maximum absorption is there in the jejunum. Fine. Maximum absorption will be there in the jejunum. Here moderately vascular it is. Ileum is your moderately vascular. So obviously absorption is also not maximum. Moderate absorption is there. Fine. Moderate absorption is there. And this ileum, it's the largest part of small intestine. It is the largest part of small intestine. So in the small intestine, these are the three parts that we have: duodenum, jejunum, ileum. And you know that in small intestine, what mucosa is going to do? Mucosa will form finger-like enfoldings, and we used to call it as villi. What is the role of these villi, bache? To increase the surface area for the absorption of the food. Because your maximum absorption will be there in the small intestine. So for the maximum absorption of the food, these villi are required. Clear, bache? Clear, bache? Now when you talk about the duodenum, here the villi, they are small. right? In jejunum and ileum, your villi are long and even they are pointed. Do you know that? Even they are long and even they are pointed, right? Even they are pointed. Clear, bache? Clear, clear, clear? Yes. So what we have in small intestine, we have the great length of intestine, villi are there, right, bache? So that's what you need to remember. That's what you need to remember. And these intestine, as I said, bache, right, these villi, as I said, they are maximum for the absorption of the food. They are increasing the area for absorption. Clear? They are increasing the area for absorption. This is about the villi. Now, bache, when you look at the cells present here you will see brush bordered epithelium. No doubt these cells are columnar, but they are brush bordered, right? These cells are what? These cells are columnar. I hope you know the meaning of columnar cells. Tall and cylinder, having nucleus present at their base, right? So no doubt we see that mucosa is forming the infoldings there. Okay, mucosa is forming the infoldings there. The mucosa is forming villi. So as I said, villi, they are small and even they are blunt in duodenum, but these villi, they are long and pointed in jejunum and ileum, right? And when you look at these villi, when you observe them under the microscope, you are going to see that brush bordered epithelium is there. What type of epithelium is there? Brush bordered. What is the meaning of having brush bordered epithelium? Brush bordered means the cells, right? The cells are having small microvilli. Okay, the cells are having these small finger-like microvilli. So in short, all the setup is for increasing the area of absorption, right? All of it is for increasing the area of absorption. Clear, bache? Clear? Right? Why all that things are there? For increasing the area of absorption. So brush bordered columnar epithelium is there. None. Brush bordered columnar epithelium is there. No doubt it is absorptive, but as well as it is secretory also. Clear? It is absorptive as well as it is secretory also. Clear, bache? Sure? Yes. Is it sure? Done. So now, other than this, in the small intestine, what will you see? In ileum, in mucosa, basically in mucosa layer of ileum. और mucosa layer में भी बच्चे middle layer of ileum, right? It contains white patches, of lymphoid tissue. Basically in ileum you will see white patches of what? Lymphoid tissue, right? Of lymphoid tissue, that is known as Pierce patches. So what is it? It is lymphoid, so it is meant for the protection, or obviously. So in the NEET examination they can ask you where these Pierce patches are present. So it is important MCQ. Clear, bache? It is important MCQ. Done. So they can ask you about the Pierce patches. Fine, bache? They can ask you about the Pierce patches. Okay? Sure. Now, bache, when you talk about the small intestine, we even see, like, let's say these are the villi. Okay? So at the base of the villi, right, we will see some tubular glands. Okay, at the base of these villi, we are going to see some tubular glands. Do you know what we call them as? We call them as crypts of Leber-Kuhan. What are they? They are crypts of Leber-Kuhan. What is it? It is the crypts of Leber-Kuhan. Write down. Write down. Pierce patches is not out of NCERT. It is important. Write down. Okay. So these are crypts of Leber-Kuhan. Write down. Then I will tell you the function here. And please be quick. Crypts of Leber-Kuhan. They are simple tubular glands present at the right? They are found between the bases of villi. Okay, as I said, they are what? They are the tubular glands present at the base of villi, right? Now, along with the small intestine, now we are even going to cover the intestinal glands, and at the end we will discuss about this salivary gland, liver, and pancreas and gallbladder. Okay, so let's finish this stomach, gastric glands, and the this intestinal glands here only. Clear, bache? Clear, clear, clear. So they are present at the base. Okay. Okay, and these crypts of Leberkuchen, these tubular glands, of course they have some cells, like Penneth cells are there, right? Enteroendocrine cells, also known as argent Tiffin cells are there, right? Bache, goblet cells are there, some another enzyme secretory cells are also there. Clear? Some other enzyme secretory cells are also there. Clear, bache? But before that, what you need to remember, see, I told you about crypts of Leberkuchen present at the base of villi, simple tubular glands they have, and they have some cells, right? So as I said, which cells are there? Penneth cells are there, enteroendocrine cells, also known as argent Tiffin cells, and some other enzyme secretory cells are also there. Some other enzyme secretory cells are also there. Clear? But here, one more thing. I'm repeating that point again, bache. I'm repeating that point again, that in submucosa of duodenum, I told you already, but again I'm telling you, Brunner's gland is present. Done? Which gland is present? Brunner's gland is present. Clear, bache? In submucosa of duodenum, Brunner's gland is present, and when it comes to the Brunner's gland, they secrete non-enzymatic, like it is not going to secrete anything where enzymes are present. So it will secrete non-enzymatic, right? It secretes non-enzymatic intestinal secretions, and of course that secretions are alkaline in nature, means they are basic. Fine, fine. So Brunner's gland, they will pass their secretion here to the base of this crypts of Leberkuchen. Clear, bache? To the base of this crypts of Leberkuchen. Done? Is it clear? Sure? Sure, sure, sure? Sure? Done, bache? So okay, so what is the function of these Penneth cells? Can you tell me? What is the function of these Penneth cells? Can you tell me? What is the function of these Penneth cells? Bache, it secretes lysozyme. So ultimately in your intestinal secretions, right, lysozyme and other antimicrobial agents are there. Okay, and other antimicrobial agents are there. Fine. So Penneth cells, they secrete lysozyme. Lysozyme is what? It is antimicrobial. Fine. It is antimicrobial. What is it? It is antimicrobial. Clear, bache? Clear, bache? Right. So the secretion of these Brunner's gland, fine, and the secretion of these Penneth cells, together they are making intestinal juices. And let me tell you, bache, intestinal juices, small intestine, let me write it here. Secretions are intestinal juices, also known as, these intestinal juices are also known as succus entericus. They are also known as succus entericus. Clear, bache? It is also known as succus entericus. Clear, clear, clear? Done. So the secretion of Penneth cells and the Brunner's gland, together they are making the secretion of small intestine. Clear? Clear, clear, clear? Sure? Sure. So next is your enteroendocrine gland, right? Next is what? Enteroendocrine gland. So obviously it will release, also known as argentifin cells, it will release hormone. What will be the function of? What will be the function of enteroendocrine cells here, bache? It will release hormone. Just say, let's say secretin hormone. It is the first hormone discovered by the scientists. Do you know that? Secretin hormone. It is the first hormone discovered by the scientists. Right? It is the first hormone discovered by the scientist. Clear, bache? Sure? Sure? Done. And then enzyme secretory cells, obviously they will release different types of enzymes. So if I have to tell you about the composition of intestinal juices, so let me write it here. Composition of intestinal juices. So it will include intestinal juices, also known as succus entericus. This is important to remember. First of all, intestinal juices, they are alkaline in nature, right? What is the pH of intestinal juices, bache? They are alkaline in nature. So pH is near about 7.8. What is the pH here? It is near about 7.8. If roughly I talk about the daily secretions, okay, if roughly I talk about the daily secretions, so it is near about 3 liters. Right, it is near about 3 liters. And if I talk about the enzymes which are present there, so you will see, very important, it is enterokinase is there, which is a non-digestive enzyme, but very important it is the non-digestive enzyme. The name of the enzymes, it is very important, please note down. So what do we have in the intestinal juices? First of all, you have to remember pH is 7.8. Daily secretions are near about 3 liter. So enterokinase is there, bache. Enterokinase is there. And other than that, your disaccharidases, like the enzymes which will help in the digestion of disaccharides, like your maltases, sucrases, also known as invertases, right, lactases. Clear? These enzymes are present. So enterokinase is there, Jyoti. Disaccharidases, like your maltase, right, like your, what is it? It is your maltase, your sucrase, your lactase, and even intestinal lipases are also there. Your nucleotidases, composition is important. Nucleosidases, hope you know the difference in between nucleotides and nucleosides. Even your dipeptidases, these are the enzymes which are present there in the intestinal juices. So please note down. Clear? Clear? Dipeptidases, disaccharide, okay, your lipases, nucleotidases, nucleosidases, they all are present there. Clear? They all are present there. Done, bache? That's important. Please note down. Done? Sure? So this is about the secretions of the small intestine. Okay, it's all about the secretions of the small intestine. So about the Peyer's patches, they can ask you in the paper about the secretions, about the position of Brunner's gland, right? And what are the crypts of Leberkuchen? So the tubular glands present at the base of villi, distributed throughout the small intestine. Clear, bache? Distributed throughout the small intestine. Okay. Done? Any doubt? Any doubt? Let me show you the diagram. See this? Small intestine. Okay. No, here firstly let's have a look of layers. It is not the small intestine. Let's have a quick revision. So see, mucosa, innermost layer, surface epithelium, lamina propria, and muscularis mucosa is there. So even in your mucosal layer, muscles are present, right? Muscles are present. Then see, submucosa, and this is the muscularis externa and serosa. So this part is of stomach, you guys can see, right? It is the histology of stomach. And as I said, in stomach, along with longitudinal and circular muscles, oblique muscles are also there. Any doubt? Any doubt? And look at the cells here which are present in mucosa, parietal cells, okay, the chief cells, your enteroendocrine cells, where we are mainly talking about the G cell. Doubt? I hope there is no doubt. Okay, so this is a structure from your book again, from NCERT. So you guys can see it is the section of small intestine. So look at the blood supply. Arteries, veins are present, and these are that crypts of Leberkuchen. These are that crypts of Leberkuchen. So tubular glands present at the base of villi. So in the villi, right, bache, no doubt, here you will see the blood supply, but here we will also see the lymph vessel, right? Villi are also receiving these lymph vessel, and this large lymph vessel is lectile. What is it? It is lectile, and we will see its role in the absorption of the fat. Fine, we are going to see its role in the absorption of the fat, bache, lectiles. Clear? Clear? So the diagram is important. So this yellow colored is your lymph vessel. It is involved in the fat absorption. Fine, lectiles. Done. So look at this. Again, it is from stomach region. So just have a look of this part, how these cells are present here. Right, bache, these are the gastric pits basically. See? Surface mucosa cells are present there. Mucosa neck cells are also there. Your parietal cell, chief cell, and G cells are there. So hope, guys, so please observe this diagram carefully. Right, observe this diagram carefully. And see, these are the secretions of your stomach. Pepsinogen will get activated from pepsin, HCl is there, mucus is there, intrinsic factor is there. Done. So it is good for your quick revision, but yeah, I will share the PDF and you guys can revise this part quickly. Okay. Chalo. So the next part is our large intestine. So we have small intestine. See, we have stomach. Stomach will lead into small intestine, which will further go into the large intestine. So I told you already, bache, that in the small intestine you have D, J, I. That's what you have. You have D, J, I. This is what you have in small intestine. It's DJI, duodenum, jejunum, ileum. So now this ileum will further leads into the large intestine. And in large intestine, bache, we have cecum, colon, rectum. These are the three parts that we will study here in the large intestine. Small intestine, duodenum, jejunum, ileum. Large intestine, cecum, colon, rectum. Cecum, colon, rectum. Clear, bache? It's cecum, colon, rectum. So ultimately, it means that your ileum of small intestine will lead into the cecum of large intestine. Clear, bache? So at the junction of this ileum and cecum, you will find ileocecal junction. What will you see here? You will see ileocecal junction. You will see ileocecal junction. Clear? So cecum, then colon, then rectum. Cecum, then colon, then rectum. Fine? Fine? So, but ultimately, why this? See, small and large intestine. See, ultimately it is related to the diameter. Just when we are saying small intestine, its length is more than large intestine. It's just that its diameter is smaller. Okay, its diameter is smaller, right? And when you talk about the large intestine, its length is not that small, but here also its diameter is larger. What is larger there? Here the diameter is larger. Okay, so now just look at this part, right? The cecum, colon, and rectum. Can you see this part? Can you observe this part? Yes. Can you observe this part? So when you talk about the cecum, it's a blind sac. Okay, the cecum is what? It's a blind sac-like structure. Means blind means closed from one end, right? It's a blind sac-like structure. It is closed from one end. What is the cecum, bache Srinath? It is closed from one end. Are you getting it? Yes. So cecum is nothing. It's a small blind sac. And here in this cecum, in this blind sac, it hosts many microorganisms. right? It hosts many microorganisms. So actually बच्चे, in our digestive system, in our alimentary canal, we have the gut flora. What is the word? We have the gut flora. Gut flora means we are talking about the friendly microorganisms here. Friendly bacteria we are discussing basically. Or you can say that friendly microorganism. For it, we are using the word gut flora. Just say you even see the ads, right? Probiotics and all. It is very good for digestion. Why? Because they will help in the growth of that flora of the digestive system of the alimentary canal. Are you getting my point? So when you are talking about the cecum, it's the blind sac. It hosts many microorganisms. Right, bache? Right, bache? And if you will see that this finger-like, this worm-like outgrowth is also there from the cecum. Can you see that? Can you see that? Just below the ileocecal opening, a worm-like structure is also there, right? Just below ileocecal opening, worm-like outgrowth is there, which is known as vermiform appendix is present. Clear, bache? It is vermiform appendix is present. Can you see this? Even in the diagram here, this part. So just below the ileocecal opening, you will see, right? Vermiform appendix. This one, vermiform appendix. Clear, bache? What is it? It is the vermiform appendix. Done. And this vermiform appendix, bache, it's a vestigial organ. In the last class, we were talking about the molars, that all the last molars, 4 last molars, they are vestigial. 4 last molars, they are vestigial. So what is the meaning of vestigial organs? Vestigial organs are the ones, right, which are not functional in our body, right? They used to be functional in our ancestors or in related species. So they have the evolutionary significance, right? By studying these vestigial organs, we can say that yes, we are related to that particular organism or this particular organism, but they do not play a significant role in our body. Clear, bache? They do not play any significant role in our body. So that is why we are saying they are the vestigial organ. Here, even it is also believed that vermiform appendix is having some lymphoid tissue. So if lymphoid tissue is there, so yes, sometimes we say that it is fighting with the infection. What is it doing? It is fighting with the infection, right? It is fighting with the infection, right? But ultimately it is vestigial. And sometimes there is an inflammation here in this vermiform appendix. It is known as appendicitis. What is it? It is known as appendicitis, right? What is it? It is appendicitis. Okay, so obviously surgical removal will be there then. Surgery needs to be done. Fine? Fine. Sure? Sure? And next to it, you guys can see here, see, cecum, then colon is there. So this is ascending colon, this is transverse, this is your descending, and this is sigmoid colon. So colon is having 4 parts. It is the middle part of the large intestine, ascending, transverse, descending, and sigmoid colon. Right? Ascending, transverse, descending, and sigmoid colon. So mainly in the large intestine, there will be the water absorption. Right, mainly there is the water absorption, and even some drugs, some medicines get absorbed here in the large intestine. So ultimately that's how it works. So finally this colon will lead into the rectum, and that rectum will lead into the anal canal. Clear, bache? That rectum will lead into the anal canal. Clear, bache? So colon continues in the rectum, right? So this rectum is basically a storage chamber for feces, right? Our fecal material, waste material. Storage chamber of feces. So our fecal material, our waste material will get stored here in the rectum. Clear, bache? It will get stored here in the rectum. Clear? So this rectum will further pass into anal canal, and this anal canal will open as anus. Clear, bache? Right? Then comes what? Then comes the anal. So anal canal opens outside by It opens outside by anus. Clear, bache? So this anus is controlled by anal sphincter. This anus is controlled by anal sphincter. So we have two anal sphincters, right? How many anal sphincters do we have? Two anal sphincters are present. Fine, bache. Two anal sphincters are present. Your intrinsic anal sphincter, extrinsic anal sphincter, right? So there are two openings, two sphincter. So extrinsic is voluntary. Voluntary means you can control it with your own will. So intrinsic is involuntary. Fine, intrinsic is involuntary. So bache, it's understood. Like if extrinsic anal sphincter is voluntary, it's understood, right, that it is having the skeletal muscles, because skeletal muscles are the voluntary ones. And when it is involuntary, of course, here we have the smooth muscles, because smooth muscles are involuntary. We cannot control their function with our will. Clear, bache? We cannot control the function here with our will. Done? Yes. Is it clear? So this is all about the, right, about the small intestine, large intestine, rectum and colon. Now, next part that we need to discuss is your glands. So we will start with the salivary glands. We are done with gastric and intestinal. So after salivary glands, we will talk about the liver, pancreas, even we will talk about the bile juices, right? I will make the duct tree for all of you, right? And then finally when it will be done, we will start the physiology of digestion. Clear, bache? So if up to this part, if there is any doubt, do let me know, right? Next we will start with the, yes, we will start with this NCERT reading, right? Firstly we will read the NCERT, then we will start the next part. So if there is any doubt, please let me know. Do you have any doubt? No? No? Sure? Okay. So see, walls of alimentary canal from esophagus to rectum, they have 4 layers. Now you know about these 4 layers: serosa, muscularis, submucosa and mucosa. Direct MCQ. Clear, bache? It's a direct MCQ. Next, serosa, the outermost layer and made up of thin mesothelium. When do we use the word mesothelium? When it is made up of mesoderm also, and it is actually the epithelium of the visceral organ. Epithelium of the visceral organ. Directly this line can be asked as a question, right? So muscularis is having smooth muscles. So if smooth muscles are present, so we know that there will be peristalsis, that is rhythmic contraction and relaxation, right? It's a rhythmic contraction and relaxation. Fine. So here you know we have ICOL. Clear? It's ICOL. And as I said, oblique muscle layer is also present in some regions. So you know it is present in the stomach. Clear? It is present in the stomach. So submucosa is having loose connective tissue like your areolar tissue, right? It is also containing nerves, bloods and lymph vessel. So in duodenum, glands are also present in submucosa. Can you name that gland? Yes. Can you name that gland? You know that that is your Brunner's gland. Another MCQ. Fine. That is your Brunner's gland. So, bache, ultimately in plant physiology and in human physiology, we need to decode the NCERT. Do you know that what we have to do? We need to decode the NCERT. Clear? Clear? Okay. So, innermost layer lining the lumen is the mucosa. So, it is having irregular folds, that is rugae in the stomach, and it is forming villi in small intestine. Clear? It is forming rugae in the stomach and villi in small intestine. Clear, bache? Clear? So villi, you know that cells are also having microvilli. So it is brush bordered epithelium, right? Why? Because of the presence of these microvilli. Clear, bache? So it is ultimately for increasing the surface area for the absorption. So these villi are having network of capillaries, large lymph vessel called the lectile, and mucosal epithelium is having goblet cell which will secrete mucus which will lubricate the food. Okay, so mucosa also forms gland in stomachs and in crypts in between the bases. I hope it is clear to you all, right? I hope it is clear to you all. That's the part that we have already covered, bache. Okay, so digestive glands में buccal salivary glands we need to start. Rest everything we have already covered, right? So we will start with the salivary glands. We will talk about the liver. We will talk about the pancreas. Okay, and then the physiology of digestion and the disorder. That's it. Fine. So do you need break or can we go like this? Chalo, I'll give you break from 7 p.m. to 7:15. So it will be a tea break, bache. So see if you guys will support, we can finish this chapter today itself. Okay, if you people will support, we can finish this chapter today itself, and Sunday we can go for the MCQ practice. Okay, on Sunday we can go for the MCQ practice. Done? So 7 p.m. to 7:15 p.m. break time. And then we will start the next part. We'll start the next part. Hi everyone, welcome back. Welcome back. Chalo. So now let's start the digestive glands, okay? Fine. So here you guys can see that when it comes to the digestive glands, you know that they are associated with the alimentary canal and together they are making the digestive system. So here we will talk about the salivary glands, the liver and the pancreas. So firstly, let's start with the salivary gland. So guys, here you guys can see the pictures as well. You can see this picture, right? So we have three salivary glands: parotid, submandibular, sublingual. How many salivary glands do we have? We have three salivary glands. So let me write down. But if you talk about the salivary gland, you know that we have to follow the NCERT, right? What we have to do? We have to follow the NCERT. And in NCERT, it is clearly mentioned that salivary glands, they are present outside the buccal cavity. Okay, where are they located? They are located outside the buccal cavity. This is your NCERT language. So they are not present within your buccal cavity. They are present outside the buccal cavity. Now when we talk about these salivary glands, they are exocrine glands. What type of glands are they? They are exocrine glands. What is the meaning of exocrine gland? That they have the ducts, bache. Clear? So now imagine, like let's say this portion is the buccal cavity. Okay. So let's say salivary glands are present here. So these salivary glands, with the help of their duct, with the help of this tube-like structure, okay, with the help of this duct, with the help of this tube-like structure, they will pour. Right, they are going to pour their secretion into the buccal cavity. Right, they are going to pour their secretion into the buccal cavity. Are you getting my point? Yes. So what do we have? Three salivary glands are there located outside the buccal cavity. They are exocrine glands. They have the ducts, they have the tube, and with the help of that ducts, they will pour their secretion into the buccal cavity. That's what you need to understand. So three glands are parotid, located below the ear. Can you see that? It is somewhere near to your ear, below the ear. Here it is present. In the cheek region, here it is present. Clear, bache? So it is not up, right? Right. So it is located below the ear. Clear, bache? And if you will talk about the parotid gland, only one pair of parotid gland is present. Can you see this? And now, bache, do you know that this duct of parotid gland, this is known as, this is what you need to remember. See, we have three salivary glands, right? Parotid, submandibular, sublingual. So MW. Okay, keep it in your mind. MW, right? You know na, M and W. W is just like you're writing M in an opposite manner. It is just like that, na? So M means mandibular, W means Wharton's, right? M means mandibular and W here, it means Wharton's. It means Wharton's. So submandibular gland, it is having a duct. The duct of the submandibular gland is known as Wharton's duct, right? It is known as Wharton's duct. Now when it is PS, right? Parotid gland, P for parotid gland, and S is your Stenson's duct. Which duct is there, bache? It is your Stenson's duct. Clear? And here you have sublingual. The trick here is LR. So here we will talk about the duct of Ravenous. Done. So no doubt it is not given in NCERT, but yes, bache, this question can be asked in the paper. For human physiology, we have to go beyond NCERT. So that's what you need to remember. So we have three salivary glands, basically three salivary glands are there. So parotid located below the ear, one pair is there and its duct is known as Stenson's duct. The duct is known as Stenson's duct, and parotid is the largest, right? It is the largest salivary gland, bache. So parotid will release saliva plus salivary amylase. These are the secretions which are produced by this parotid gland. right? So what will it do? It will secrete saliva plus salivary amylase, and salivary amylase is also known as ptyalin, right? It is also known as ptyalin. So basically here we are talking about what? We are talking about the saliva. Saliva, you know that it is present in our mouth. So obviously in its composition we know maximum amount is of water, but there are other things also. So who's going to synthesize, who's going to form that saliva? These glands. So we have 3 glands and 3 of them are present as the pair. So parotid gland is there, right bache? Your submandibular gland is there, your sublingual is there. So parotid located below the ear near to the cheek region, 1 pair. Stenson's duct, saliva plus salivary amylase will be synthesized by this parotid. So you can write down maximum salivary amylase is formed by parotid gland, right? Maximum salivary amylase is formed by the parotid gland. Is that clear? Is that clear? That's important, bache, right? So it is the largest salivary gland. Okay, it is the largest salivary gland. Done. And yes, you are right. The viral infection, it's viral infection. You have to write down the name of the virus in the comment section. So viral infection of this parotid gland is mumps. What is it? Mumps. Viral infection of this parotid gland is what? It is mumps, and you will mention the name of that virus in the comment section after it. Okay, done? Sure bache. So next is your submandibular, right bache? Submandibular is also written as submaxillary. Do you know that? Submandibular, it is also written as submaxillary. Actually this gland is present at the junction. Can you see this? It is present at the junction of the jaws, and it is more towards the lower jaw. Okay, you guys can see here, right? You people can see here submandibular duct, right? Submandibular gland, submandibular gland. So it is present at the junction of jaws. Clear? So we also use the word submaxillary gland for it. So it is present at the junction of jaws, and it is more towards lower jaw. Okay, it is more towards lower jaw. Clear, bache? So it is present at the angles of this, right? At the angles of jaw. Clear? So again, 1 pair, right? And MW. So the name of the duct is Wharton's duct. What is the name of the duct? The name of the duct is, yes bache, the name of the duct is Wharton's duct, right? So this is the gland which will only secrete saliva. So it is the gland which it secretes maximum saliva. Okay, it secretes maximum saliva. So it is not going to form that salivary amylase, or if it will form, it will be of very less quantity, but it will secrete maximum saliva, right? It will secrete maximum saliva. Okay, bache, done. So next is your sublingual. So now when the word is sublingual, lingual word is associated with the tongue, isn't it? Lingual word, it is associated with the tongue. So it is present below the tongue. Okay, so if I talk about its location, it is present below the tongue. Okay, and it will have many ducts, right? It will have many ducts. So that are ducts of Ravenous. It's LR. Okay, it's duct of Ravenous. What is it, bache? It is the duct of Ravenous. Clear, bache? Clear. So these are the smallest, right? So smallest salivary duct, and obviously it will also produce saliva only. Moderate amount of saliva will be formed by this particular gland. So it is having the smallest ducts. Clear, bache? Clear? So you guys can see the positions here. I hope it's clear. See this diagram. So see, so many ducts are there, right? Duct of Ravenous. Duct of Ravenous. This is your Wharton's duct, the largest salivary duct. And this one is your parotid gland and parotid duct. As I said, it is your it is Stenson's duct. Right, it is your Stenson's duct. Done. So students, you can revise it from this part, right, bache? So this is about the salivary gland. So obviously salivary gland is going to form the saliva. So what will we see in saliva? Can you just tell me the composition of saliva? Can you just tell me the composition of saliva? Yes. Please tell me the composition of, yes, the composition of saliva, and do it quickly. First of all, bache, when it comes to saliva, the pH is near about 6.8. Right? What is the pH here? What is the pH here? The pH is near about 6.8. And if I talk about its daily secretion, so it is a 1 to 1.5 liter. Okay. If I talk about the daily secretion, so again, it is also 1 to 1.5 liters. Clear, bache? So maximum is water. Water is near about 99.9% is the water here. Right? 99.9% is the water here. Or simply you can say that it's 99.5%. But okay, you can simply keep it 99%. Right? 99% is what? 99% is the water here. And along with water, some salts, some ions are present. Right? Like you will also see the sodium ions. You will even see the chloride ions. Right? Some thiocyanates are also present. Some thiocyanates are also present. Right? So sodium, chloride, bicarbonate, mucins, right? All of them are present here. Clear, bache? All of them are present here. So sodium, potassium, chloride, etc. Other than this, bache, thiocyanates, right? They are antimicrobial. Okay, when it comes to the enzyme lysozyme is present there. The enzyme salivary amylase, also known as ptyalin, is present there. Right? Salivary amylase, also known as ptyalin. Ptyalin is present here. What is present here? Salivary amylase, which is also known as ptyalin. So lysosome, you know that it is antimicrobial. What is it? It is antimicrobial. Clear, bache? So it is present in your saliva. Done? Done. So thiocyanates, these salts, they are present. And let me tell you, bache, these chloride ions are important for activation of your salivary amylase. Right? It is important for activity of your salivary amylase. Remember this. Done? It is important. Chloride ions are important for the activity of your salivary amylase. So thiocyanates, right, bache, your bicarbonates, all of them are present here. Clear? All of them are present here. And do you know, other than this, yes, other than this, what do we have here? Other than this, what do we have here? Do you know that? Urea, some amount of waste is also there, na? Some amount of urea, uric acid, right? It is also present. It is also antimicrobial. So all of these things are going to keep a check on the microorganisms in the buccopharyngeal cavity. Clear, bache? In the buccopharyngeal cavity. Clear? And yes, in the saliva, IgA is present. This is important question. Fine. IgA. IgA means your immunoglobulin A is present. Like even in the tears, even in the saliva, antibody, immunoglobulin A means antibody A is present. Right? Antibody A is present here. Done, bache? What is there? Antibody A is present. Now, bache, how the saliva secretions will be stimulated, or you can say that what is going to increase and decrease the secretions of saliva. Now you know that when we look at a food item, right, even if you will think of it, right, imagine you are thinking of like in my case, paranthe, theek hai, in your case it might be pizza, burger, anything it can be, right? If you will even start thinking about it now, you know that the saliva production will increase. So saliva, it is helping in the digestion. How? You are eating the food, right? Teeth are going to masticate it. The food will get mixed with the saliva. So obviously it's a kind of lubrication as well, right? And even that saliva is having the enzymes, mainly the salivary amylase. So this salivary amylase and ptyalin, it's very important for the digestion of the carbohydrate. So that is why we used to say that 30% of the starch is digested in buccal cavity, right? 30% of the starch is digested in buccal cavity, right? 30% of the starch. Are you getting my point? Right, that's important to remember. So obviously nerves are responsible for its secretion, for inducing and inhibiting its secretion. So this is what you should note down. As of now, you must be thinking that, ma'am, it is not given in digestion and absorption, but it is there in nervous control, right? So you better write it here. So here you guys can write that sympathetic nerve fibers, right? Sympathetic nerve fibers decreases the secretion of saliva. That's important. They decreases the secretion of saliva. And parasympathetic fiber, because their main role is in rest and digest, right? So parasympathetic nerve fiber, it increases saliva secretion. Fine, it increases saliva secretion. Clear, bache? Sure? Are you sure? Yes? Tell me, bache, any doubt? Is it clear? Sure? Done? Are you sure? Right? So now, just look at this saliva. See, we will revise 2 topics altogether. So now when it comes to saliva, you know its composition. So you know only one thing, and that is the salivary amylase is present. But here we are not talking about the proteases. We are not talking about the lipases. Luke, bache, we are not talking about the proteases. We are not talking about the lipases here. So you can simply say when it comes to the mouth region, when it comes to the buccal cavity, in the buccal cavity, what will be there? There will be the digestion. What will be there? There will be the digestion of carbohydrates. Right, there will be the digestion of carbohydrates, and as I said, 30% of starch will be digested here. Okay, so the another name of salivary amylase, very important, and that's ptyalin. And let me tell you, bache, these are the alpha amylases. They are not the beta amylase, it is the alpha amylase. Alpha amylase, right? Chalo, let me explain this. I hope you know that we cannot digest the grass, right? Uncooked grass cannot be digested by our body. Are you aware of it? Uncooked grass cannot be digested by our body. Are you aware of it? I hope you guys are. Right. Now in grasses, you know that their cell wall, it contains the cellulose. Right. Now when you are talking about the cellulose, cellulose is a unbranched, right? It's an unbranched beta polysaccharide. It's a polysaccharide, no doubt it is made up of glucose, but it's a beta polysaccharide, right? It's a beta polysaccharide. Do attend the biomolecule lecture. Beta polysaccharide means here it is having beta 1,4 linkage. Which linkage is there in the cellulose? Beta 1,4 linkage is there, right? So now the enzyme that are present in our body, they are the alpha amylases. Like they can digest the polysaccharides which are having this alpha bonding, not the beta bonding. Clear, bache? Not the beta bonding. So all that things, right? Because the cellulose is a beta polysaccharide, so it is not digested in our body. We do not have the cellulase in our body. We have the alpha amylases in our body. Clear, bache? We have the alpha amylases in our body. So as I said, 30% of the starch will be digested in our body. Clear, bache? Clear, bache? That's the point. So now after this buccal cavity, although we have already discussed the gastric secretions, right? Right? So let's have a quick revision of that. So we have already discussed the secretions of the gaster, secretions of the stomach. But again, let's have a quick revision. Okay, let's have a quick revision of that. See, stomach or gaster. So you know that in the stomach, this is what we have. It's mucous cells, parietal cell, oxyntic cell, chief cell, also known as zymogenic cell, and peptic cell. So I hope you all are aware of it. Right? So now you can even relate this part, isn't it? You can even relate this part. Now, in the stomach, in the buccal cavity, you have the bolus. With the help of peristalsis, this bolus will pass to the stomach. Now, in the stomach, you know that there will be no digestion of the carbohydrate because amylases are not there. They are non-functional there. Why? Because in the stomach, the conditions are acidic. What type of conditions are there? In the stomach, the conditions are acidic. And have you seen that? In the case of the salivary amylase, in the saliva, pH is 6.8. Now in the stomach, the pH is near about 2. Acidic pH is there. So even that bolus having saliva, having salivary amylase will not be functional there in the stomach. Right? The carbohydrates will not be digested there in the stomach. Right? You know, na, HCl, it kills microorganism, inactivates the salivary amylase tylen. So I hope now you can understand this point. And as I said, it helps in the activation of inactive proteases. And moreover, bache, this HCl, it converts. Do you know that? It converts Fe3 positive to Fe2 positive. And this Fe2 positive is the absorbable form in our body. Right? This form of iron can be absorbed in our body, right? HCl is also playing the role here, right? So that's how it works. That's how it works. Clear, bache? So next that we need to discuss is what? We need to talk about the liver, right? So now see, one by one we are discussing. Buccal cavity done, salivary glands done, esophagus done, stomach done. Stomach is located towards the left side of the abdominal cavity. And now the next that we are going to discuss is your, yes, bache, the next that we are going to discuss is your quick liver. Done? It is your liver. Okay. Do you know, bache, liver is the largest gland of our body, right? Liver is the largest gland of our body, not second. It is the second, largest gland of our body. So in the paper, read the question carefully. If they are asking largest gland of the body, then it's liver. But if they are asking largest endocrine gland of our body, then it will be thyroid. Clear? Then it will be thyroid. So liver is what? It is the largest gland of our body, bache. It is present on the right side of the right side of abdominal cavity. Right side of abdominal cavity, just below, okay, just below the diaphragm. Clear? Just below the diaphragm. Now you know about the diaphragm, isn't it? Now you know about the diaphragm. Clear, bache? Clear, bache? So when it comes to the liver, you know that liver is, yes, can you tell me about the weight of the liver? Yes, bache, can you just tell me about the weight of the liver? 1.22? Yes, it is 1.22, 1.5 kg. Okay, it is 1.22, 1.5 kg. Kg or G? Yes, it's kg or G? It's kg, kilogram in adult human. Fine. In adult human, right? So generally, no doubt, bache, we say that liver is having 2 lobes, but actually they are 2 main lobes, right? What do we have? We have 2 main lobes. Here you people can see just below the diaphragm it is present. This is your right lobe of liver. This is your left lobe of liver. But actually in the liver there are 4 lobes. Okay? Okay. So liver may, no doubt, there are 2 main lobes. Your right lobe, the largest one, and the left lobe. But actually, do you know that liver may there are 4 lobes? There are 4 lobes, right? 2 more are there, and that is your caudate and quadrate. That is your caudate and quadrate, right? These are the minor lobes actually. right? They are present just behind this right lobe. Okay, they are very minor lobes. Fine, they are very minor lobes. So in totality, there are 4 lobes, caudate and quadrate is also there, but 2 main lobes are there, right and the left one. Clear, bache? The right and the left one. Is it clear? Right, the right and the left one. Done, bache? Done. So it is the right lobe which is the largest, right? It is the largest lobe. So it is making 5/6 of the liver, right? Remaining is your left lobe. Done, bache? Remaining is your left lobe. Is that clear? Sure? Sure. Done? So now, one more thing is there, bache. When you talk about this right and left lobe, na, they are separated by the ligament. And do you know that what is the name of that ligament? That is your falciform ligament. Of course, it's a type of connective tissue, isn't it? It's a type of connective tissue. What do we have here? We have the falciform ligament. So, right and left lobes, they are separated from each other by falciform ligament. Let me write. Right and left lobe of liver. Right and left lobe of liver separated by, they are separated by falciform ligament. So, bache, it's a ligament. Obviously, it's the example of the connective tissue. Done, bache? It is the example of what? It is the example of the connective tissue. This point is important. Please note down. Okay, this point is important. Please note down. Fine. Sure. Done, bache? Okay, so the next point is the gallbladder. There is one more sac-like structure. Do you know that? Gallbladder. It is situated just below the right lobe of liver. Do you know that? It is situated just below the right lobe of liver. So here you can see. Can you see this part? This is the gallbladder. Okay. What is it, bache? This is the gallbladder. It is the gallbladder. Actually here, you know, liver is performing different roles, right? Liver is going to do different functions. Fine. So one of it is कि liver will form bile. What will liver do? Liver is going to form the bile. We will talk about the composition of the bile as well. Don't worry. Okay. So liver is going to do what? Liver is going to form the bile. Okay. So this bile is very important for the fat emulsification and the fat digestion. Okay. So this bile, it will be stored here in the gallbladder. So what is the function of gallbladder? To store and concentrate, right? To store and concentrate the bile. You need to remember both the words, right? So its main function is to store and concentrate the bile. Are you getting it? So it is located just below this right lobe of liver, and it is also kind of pear-shaped structure, right? In some people, you know, the gallbladder, just some people, if they have the stones in their gallbladder, so after surgery, that gallbladder will be removed. So in their case, like, it is not going to harm. If gallbladder is not there in our body, it is not going to create some, you know, problem. But in that case, we cannot digest the fat properly. Okay, fat digestion will be problematic then, right? Because no doubt it is going to, its main function is to store, but to concentrate the bile as well. And this bile is very important for the fat storage. Are you getting it? This bile is very important for the fat storage. Clear, bache? Clear? So before telling you the structure of liver in detail, let me tell you about the duct tree first. Okay, let's discuss about the duct tree first. So now just have a look, right? This is your liver separated by your falciform ligament, which is fibrous connective tissue, and just below this right liver, here you guys can see the gallbladder, right? Here you people can see the gallbladder. Clear, bache? Clear? So where is our diagram? Yes, this diagram. So see, liver, gallbladder. Then can you see this pancreas here? Pancreas here, right? Can you see this? This is the liver located on the right side of the abdominal cavity. This is the gallbladder situated just below the right lobe of the liver. And here you have this pancreas, right? Here you have this pancreas. Can you see this part? Yes or no? Yes or no? Right. First of all, just look at the positions here, right? And then I will explain what is the duct tree. Okay. Then I'm going to explain that what exactly is your duct tree. Clear, bache? Clear, clear, clear. So let's finish the liver first. Then yes, we will start this part. I hope you people are making the notes. Okay. So now when we talk about the liver, okay, so now you know the presence of gallbladder as well. Fine. Now, when we talk about the liver, in the case of liver, it is the hepatic lobule. It is the hepatic lobule, which is the structural and functional unit of liver. Right? Many times students, they do not focus on the word hepatic lobule. They mark it as hepatic cell, but actually that's not the case. It is the hepatic lobule, which is the basic, right? Which is the basic structural and functional unit of the liver. So here what will you see? You will see that, some sinuses, some spaces are there where blood vessel, lymph vessels are there. You will see some cord-like structure. Like see this hexagonal kind of structure is your hepatic lobule. So this hepatic lobule will be having, bache, it will be having your hepatic, yes, it will contain your hepatic cells. Right? What will it contain? It will contain your hepatic cells. So it is going to form cord-like structure. Fine. It is going to form cord-like structure. Right? You will see the blood vessels here. So actually it is the hepatic lobule. I will show you the better diagrams. It is the hepatic lobule, which is the structural and functional unit of life. Now, one more point is there, that this hepatic lobule is covered. Do you know it is covered with what? It is covered with the glissons capsule, and again it's a very unique feature here which is present in our case, right? It's a very unique feature here. This hepatic lobule is covered with the glissons capsule. So glissons capsule is nothing, it is the connective tissue. What's that? It is the connective tissue. Write down. Right? What is this glissons capsule? It is nothing but it is the connective tissue. So it is the hepatic lobule, basic structural and functional unit of liver, having hepatic cells which are forming the cords. So lobule is covered with the connective tissue sheath, that is known as glissons capsule. Clear, bache? That is known as glissons capsule. Now here in this hepatic lobule, as I'm saying, bache, see, these individual cells are hepatic cells. These single cells are hepatic cells. For them, we also use the word hepatocytes. Fine. We also use the word hepatocytes. So just look at the structure carefully, right? See, so many hepatic cells. 1, 2, 3, 4. See, they are forming these strong, they are forming this string-like structure. Can't you see that? They are forming some string-like structure, some rod-like structure, or some cord. The right word here, right? It will be the cord. So these are that cord-like structures. Clear? These are that cord-like structures. And in between here in these spaces, what will you see in the liver? You are going to see the Kupffer cells in the liver. What will you see here in the liver, bache? You will see Kupffer cells in the liver. Which cells will be there? Kupffer cells. It is a repeated question. So Kupffer cells are the macrophages of liver. They are the phagocytotic cells, or you can say that macrophages of liver. What are they, bache? They are the macrophages of liver. Are you getting it? Are you getting it? They are the macrophages of liver. Okay? Okay? Yes or no? They are the fixed macrophages. जैसे even in the connective tissue macrophages are there, they are histiocytes. In your nervous system macrophages are there, right? That are your microglial cells, right? Glial cells, microglial cells. And even in the liver, that macrophages, that phagocytotic cells are there, and we used to call it as Kupffer cells. Fine. So this hepatic lobule is the basic structural and functional unit of liver, and it is covered with a thin connective tissue sheath, right? It's a unique feature of mammals. Thin, yes. So it is the Glissen's capsule. So what is it? It is the Glissen's capsule. So now just look at the diagrams. See this. Right, this hexagonal kind of structure. So see bache, so these are the here you will be having the central veins, and in between also you will see that spaces, and surrounding mein bache branch of hepatic portal vein and all arteries there. Or ye dekho, these spaces, these are sinusoids, right? These are sinusoids. Clear bache? Just see this. This is a very beautiful diagram. So this central vein, blood flow out of liver, right? There will be the blood flow out of liver here, and see these are the spaces, right? So see here, the bile duct is also there. So hepatic cells will form the bile, it will pass here, and then obviously it will be stored in the gallbladder. Clear bache? Clear? So see the hepatic portal veins, hepatic arteries here in the spaces, and even in these blood vessels you will see the Kupffer cells. As I said, they are phagocytotic cells. Right, bache? They are the phagocytotic cells. So this is that internal section. Done? This is that internal section. Clear? Clear? Okay. So hepatic lobule. This is the word that you need to focus. Done? Hepatic lobule, the basic structural and functional unit of life. Right? It is the basic structural and functional unit of life. Clear? Right? So it is basic, structural and functional unit of life, not life of liver. Oh. Basic unit of life is cell, na? It is liver. Done? Clear? Sure? Yes? Any doubt? That's the internal structure of your liver. Fine, that's the internal structure of your liver. Let's finish the liver part first, and then we will talk about that duct tree. First of all, let's talk about the liver, pancreas individually, about their secretions individually, and then I will show you what exactly is that duct tree. So any doubt? No doubt? Sure. Okay, so next to it is pancreas, right? I'll tell you about the ducts, but as of now just focus here. So can you see this carrot shaped gland? It is the second largest gland of the body. Right? Second largest gland of the body. Can you see it? Yes. Bache, first of all, now let's continue with the liver secretions. Let's discuss the liver secretions first, and then we will talk about the pancreas. So, what I just said, liver is going to form the bile. But liver is also having some another functions also. Can you tell me about the functions of liver? Yes. Can you just tell me about the function of liver? Can you just tell me about the function of liver? No doubt here in context of digestion, you can simply say that synthesis of bile. Okay, the synthesis of bile will be there without any doubt. It is going to do what? It is going to form the bile. It is going to secrete the bile. And what is this bile? It's a watery fluid containing some salt. Right? Write down the composition of bile as well. It's a watery fluid containing some salts. What is it? Watery fluid containing some salts, and these salts are responsible for the emulsification of fat. Are you getting it? These salts are responsible for the emulsification of fat. Right? Can you name that salts, bache? Yes. Can you name that salts? Give it a try. Right? It's very common. The first one is your sodium bicarbonate. It's sodium taurocolate. It's sodium glycolate. Right, so these are the salts which are present in the bile. Okay, these are the salts which are present in the bile. So sodium glycocolate is there, right? Sodium glycocolate is there. Sodium toro. Yes, sodium taurocolate is there. Colate. Just a minute. 8. So sodium glycocolate. Right, so these are the salts which are present in the bile. So bile contain no enzymes, right? This is what you need to highlight. Bile contains no enzyme. Enzymes are absent in the bile. So bile is having some electrolytes, watery fluid, and the salts are present there, right? And the salts are present there. And other than this, bile pigments are also there. Right, bile pigments are also there, like your biliverdin, bilirubin. Fine. So bile pigments are there, bile salts are there. Write down the name of the pigments as well. And other than this, even some amount of cholesterol is also present there, bache. Right, some amount of cholesterol is also there in the bile. And yes, the phospholipid lecithin is also there in the bile. Do you know that? The phospholipid lecithin is also there in the bile. So the composition of bile is important, right? The composition of bile is important. So it's not just only lecithin, cholesterol, other compounds are also there. So you can just write down that mainly it contains salts, and these salts are very important for the emulsification of fat. Now, what is the meaning of emulsification of fat? Breaking down the large fat globules into the smaller one. Clear? Breaking down of large fat globules into the smaller one. That is the meaning of your, yes, emulsification of fat. So this is the composition of the bile, which will be stored and concentrated there in the, yes, it will be stored and concentrated there in the, yes, bache, in the gallbladder. Clear? Clear? Sure? Sure, bache? So other than that, what is the function of the liver? Do you know that? Do you know that the liver is also helping in the carbohydrate metabolism? It is playing role in the carbohydrate metabolism. Like, okay, let me quote the example. They say glycogenesis. You know that what is the meaning of glycogenesis? Genesis means formation and it's glycogenesis. Right, genesis means formation and it's glycogenesis, formation of your glycogen, right? So in the liver, excess of glucose will be converted into glycogen, right? So glycogenesis there will be, and even glycogenolysis, right? So whenever in the blood the sugar is low, so what is going to happen, bache? The breakdown of glycogen into glucose will also be there. The breakdown of glycogen into glucose will also be there. Right, so that is why I am saying that it is playing role in the carbohydrate metabolism. Are you getting it? Are you getting it? Then comes bache gluconeogenesis. It will also be done here in gluconeogenesis. It will also be done here in this liver. So now the word is neo. Neo means new. Neo means new. Genesis means formation. Neo means new, Priyanka. Genesis means formation. So here from non-carbohydrate source, glucose will be formed. So if in the body carbohydrate scarcity is there, so from non-carbohydrate source, glucose will be formed. Clear, bache? This is suppose if we are converting fat into carbohydrate, if we are converting proteins into carbohydrates, so they are the non-carbohydrate sources which are forming the glucose. So gluconeogenesis will also be done here in the liver, right? So the main function here as per the digestion, it is the formation of bile, then carbohydrate metabolism is also there, bache. So it is also going to, right, it is also going to help in the urea formation. So you can write down deamination and urea formation will also be done by liver. Okay, deamination and the urea formation will also be done by liver. Deamination and urea. See, I think one thing you all know that ammonia is very toxic. Ammonia is a highly toxic nitrogenous waste. It is a waste which requires a lot of water to get out of our body, right? In the form of ammonium ion, it usually gets diffused from the body of the aquatic organism. So in our body, there is the formation of the ammonia, but that ammonia will be converted to urea, and it will also be done in the liver, right? In liver, bache, the ammonia and your carbon dioxide both, they will be converted to urea. Ammonia plus CO2, it will be converted to urea. So in the liver, there is the urea cycle, also known as ornithine cycle, right? Urea cycle and ornithine cycle is there in the liver, and it is going to convert ammonia and carbon dioxide into the urea. Clear, bache? It is going to convert that ammonia and carbon dioxide into the urea. Clear, bache? Clear, bache? So deamination will also be done, right? So deamination and the urea formation will be done in the liver. It is also one of the most important functions of the liver. Clear, bache? Clear, clear, clear? Yes? Okay, so there are many functions of liver. Liver is a very important organ. Jaise liver stores your fat soluble vitamins like your A, D, E and K. Do you know that it is going to store your fat soluble vitamins, that is your A, D, E and K, and even vitamin B12 is also stored in the liver. Liver purifies blood. Do you know that liver purifies blood as well? Like here it is having the Kupffer cells. So when blood will flow from that liver cells, from that hepatic globules. Kupffer cells will destroy the foreign particles. Clear, bache? Kupffer cells are going to destroy the foreign particle. And if you remember, liver also synthesize plasma protein. Liver is going to synthesize plasma protein as well. Clear, bache? That's important. You know, in the blood plasma, we have the proteins. So many type of proteins are synthesized by it, right? Right, bache? Right, many types of proteins are synthesized by it. Okay, so jaise plasma mein you have the globulins, not immunoglobulins, but your albumins, right? Other, the proteins which are helping in the blood clotting, like your fibrin and all, they will be synthesized by, right? Liver. So except immunoglobulins, that is your antibodies, all plasma proteins are synthesized there. So see, liver is so important, isn't it? So any other main, or it in the detoxification, liver is going to help, you know that, right? In the liver, the cells, the hepatic cells are having smooth endoplasmic reticulum that will help in the detoxification of the drugs and all. So can you just tell me any other function of the liver? Yes, bache, any other important function of liver that we are missing? Okay, any other important function of the liver that we are missing? Anything? But that's very important in the embryonic stages. In embryonic stages, your RBCs, WBCs are synthesized by liver. Initially, they are synthesized by liver. Clear, bache? That's important function. Okay, so your yes, natural anticoagulant heparin is also synthesized by liver. So you know heparin is a natural anti-blood coagulant, so it is synthesized by the liver. Some amount of heparin is also synthesized by basophils. Clear? It is also synthesized by basophils. So this is the function of your liver, bache. Right? This is the function of your liver. So blood clotting proteins are also synthesized by it. Some enzymes are also synthesized by the liver, bache. Clear? Clear, clear, clear. So that's what you need to remember. Okay, that's what you need to remember. So already I told you about the bile juices, isn't it? Bile juices. Right? So bile juices may, remember, the composition is important and pH is also important. So can you tell me the pH Of course, it is going to be alkaline. But can you tell me the pH? Can you tell me the pH? It is somewhere in between 7.6 to 8.6. Okay, it is somewhere in between 7.8 to 7.6 to 8.6. And these pigments are going to give the color, bache, right? These pigments are going to give the color. So when it is bilirubin, it will be yellow, RY, and this one will be green. Okay, so that's it, that's all. So if there is any doubt, please tell me, bache. Please tell me. Done? Sure? Sure. So bile is very important for the fat emulsification. Okay. It is very important for the fat emulsification. Done? So sodium it's taurocolate. So bache, this taurocolate and sodium glycocolate, these are the organic salts, right? So sodium bicarbonate, even simple sodium carbonate, these are the inorganic salts. So these salts are present here in bile. So bile is helping in the fat emulsification and the fat absorption, and you know, even it is helping in the fat soluble vitamin absorption. There are fat soluble vitamins also, na, A, D, E and K. A, D, E and K, very important they are. E is the beauty vitamin. K is very important for the blood clotting. So for the absorption of that fat soluble vitamins also, this bile used to play the role. Clear? So now I told you about the functions of the liver, about the function of the bile, the role of the bile as well, which is very important, the pH as well. Clear? And even another important functions of liver as well. So please, bache, please, one way or another way, it is present in your, right? Trust me, it is there in your NCERT. In the stomach, the acidic secretions will get mixed. Then that food will pass to the small intestine. So small intestine will receive everything. It will receive the bile juices, the pancreatic juices, and the intestinal juices. So bile will bile will neutralize the acidity of the chyme. Okay, it helps in the fat absorption. It helps in the fat soluble vitamin absorption. Clear, bache? So these are the main roles of this bile. Clear, bache? And even these pigments present in the bile juice, biliverdin and bilirubin, these are the excretory products. So it is also helping in the excretion. Clear? It is also helping in the excretion. Clear, bache? Okay. And do you know, sometimes, you know, the passage within the gallbladder, it gets blocked. Then we face that issue of gallbladder stones. Okay. Then we face the issue of gallbladder stones. Clear? And do you know, bache, what happened? You know, when your bile, right? Just say, let's suppose if bile is blocked, okay, if the bile passage, if its bile passage is blocked, what is going to happen? As I said, biliverdin and bilirubin, they are what? They are the excretory products, right? They will pass out. Just say our excreta is kind of yellowish. Why? All that pigments are present there, right? Now, sometimes what happened, the passage of this bile, it gets blocked. And because of that, this bilirubin, sorry. Remember this, it is RY, right? It's RY. Okay, so the amount of this bilirubin in blood, it increases. So if amount of the bilirubin in our blood, if it gets increased, because it's a yellowish pigment, it will give that yellowish appearance, right? That is jaundice. That is obstructive jaundice. So it's a very important question. Jaundice is the disorder of the liver. Liver is not functioning properly. That is why jaundice will be there, because this bilirubin amount will get increased here in the blood. So that is why the appearance will become yellowish. Clear? Skin will become yellowish, nails will become yellowish, urine will also be yellowish. Clear, bache? Urine will also be yellowish. Clear? Clear? Done? So this is about the liver, this is about the bile. Next is your pancreas. Next is your pancreas. Okay, so pancreas, bache, it is the carrot-like gland. It is also known as, you know, sweet bread. And do you know it is the second largest gland of the body. It is second largest gland of body. And this is a heterocrine gland, right? Heterocrine gland. Also, you can use the word composite gland. It is a heterocrine gland, also known as composite gland. Now, what is the meaning of heterocrine gland and the composite gland, bache? That it is having two parts. It is even having the exocrine part as well. It is even having the endocrine part as well. So you can see this here, it is present. This is a carrot shaped, right? Sweet bread, your pancreas, second largest gland of the body, heterocrine in nature, bache, means it is composite gland. It is having the exocrine part as well. It is having the endocrine part as well. Clear, bache? It is having the exocrine part as well. It is having the endocrine part as well. And this pancreas, it is divided into that head, neck, tail. It is divided into these three parts: head, neck, and tail. Clear? It is divided into three parts: head, neck, and the tail. Right? And even body is there, of course. Then only we will have the tail. No. So head, neck, body, tail. Fine. So here you guys can see. Okay. So 99% of pancreas is exocrine. Your 1% is endocrine. Right? 99% of the pancreas is your exocrine. 1% is your endocrine. Clear, bache? So exocrine part will include your acini. Right? Acini. Clear, bache? And they will form pancreatic juices. This one is going to form the pancreatic juices. And when it comes to your endocrine part, okay, so it includes islets of Langerhans. Islets of Langerhans. So here we have endocrine part, means ductless it is. It is ductless. And when you are talking about this part, exocrine part, 99% of the pancreas is exocrine in nature, right? So acini, here we will have acini. You will see some cells. Some cells are forming some tube-like structures. Okay, so this is the part which will form the digestive juices or the pancreatic juices, also known as complete digestive juices. And when bache it is islets of Langerhans, here you will have alpha cell, beta cell, delta cell, pancreatic polypeptide cells, right? Here you will have different types of cells like alpha cells, beta cells, delta cells, and pancreatic polypeptide cells. Alpha cells, beta cells, delta cells, pancreatic polypeptide cells. Clear? So pancreas, heterocrine or the composite gland, it is having exocrine and endocrine part. Exocrine part is the 99% and 1% is your endocrine part. Clear, bache? 1% is your endocrine part. That's what pancreas is. Clear? That's what pancreas is. Right? So it is having the acini. So acini are these rounded lobules. So it is the exocrine part, bache, which will form your digestive juices. Clear? Digestive juices. Done, bache? Done. So endocrine part is 1%, islets of Langerhans. There are the cells, alpha, beta, delta, and pancreatic polypeptide. Although we will discuss this part in chemical control and coordination, still I'm giving you this idea. Alpha cell will form glucagon. So it will form hormones. Your beta cells will form insulin, right? Delta cell will form somatostatin. Somatostatin, which is growth hormone, inhibitory hormone. Pancreatic polypeptide cells, they inhibit secretions of digestive juices. It inhibits secretions of digestive juices. Fine. Fine. So it is the exocrine part, right? It is the exocrine part of pancreas which will form the digestive juices, right? It is the exocrine part of pancreas which will form, yes, which will form digestive juices. So again I'm repeating, exocrine part is containing rounded lobules known as acini. known as acini. Clear bache? Known as acini. So now it is exocrine. So it means it is having the ducts. Yes, it is having the ducts. So in the case of pancreas bache, right, we will talk about two ducts. This is important, please mark it. Right, we will talk about two ducts. One is your normal pancreatic duct. Right, it will join with hepatic duct. Right, I will explain this bache. It will join with hepatic duct. Clear? This pancreatic duct will join with hepatic duct, and another is your accessory pancreatic duct. So there are two ducts. One is your accessory pancreatic duct. So it will directly open in small intestine. Right? It will directly pass its secretions to the small intestine. So basically pancreas is having two ducts. How many ducts are there? Pancreas is having two ducts. Are you getting it? Are you getting it? Yes. So please write down main pancreatic duct. One is accessory, another is main. Main pancreatic duct, it is duct of, its another name is not given in NCERT bache, but still you can write down duct of Wirsung and your accessory pancreatic duct. It is known as duct of Santorini. It is known as duct of Santorini. What is it? It is known as duct of Santorini. Clear bache? One is duct of Wirsung, another is your duct of Santorini. Okay? Done, done, done. So now, ultimately, you know that pancreas will contain pancreatic juices. What will be there in pancreas? Exocrine part will form pancreatic juices. Clear, bache? And obviously, these secretions are induced by the hormones. Okay? Pancreatic juice. Obviously, exocrine part is going to form the pancreatic juices, which are meant for the digestion. And the hormone, we have one hormone, pancreozymin, right? Pancreozymin, it is a hormone. It is going to induce the secretion of this pancreas. Let me write down the name here. It's pancreozymin, right? It's pancreozymin. It is going to induce the secretion of these pancreatic juices. Okay, and bache, these pancreatic juices, remember, we always call it as complete digestive juice. Right? Pancreatic juices are also known as complete digestive juices. Do you know why? Do you know why? Because pancreatic juices contain all the enzymes important for the digestion of all type of substrate. Right? Pancreatic juices, they contain the enzymes important for the digestion of all type of substrate. Like for the protein digestion, you have the enzymes here. For the carbohydrate digestion, you have the enzymes here. For the nucleic acid digestion, you have the enzymes here. So that is why we call it as complete digestive juices. What are we going to call it, bache? We are going to call it as complete digestive juices. So if I talk about the pH here, so it is somewhere in between 7.5 to 8.3. Again, it is also alkaline. 7.5 to 8.3, right? What is the pH here? It is 7.8 to 8.3, and the daily secretion is in, you know, it is in, you can say that it is in, it can be in between, you can say that 1200 to 1500 ml, means 1.2 to 1.5 liter, right? Right, when you talk about its secretion, so it is going to include what? It is going to include what? So total secretion will be 1 liter to 1.5 liter per day. When you talk about the secretions, and let me tell you, if pancreatic secretions are not there, if these digestive enzymes are not there, right, we cannot digest our food so easily. It will take months to digest the food then. right? It is going to take months to digest the food then, right? So pH here is 7.5 to 8.3, right? The daily secretions 1 liter to 1.5 liter. As I said, it is having enzymes for the digestion of all types of food. Jaise these pancreatic juices, they will contain pancreatic alpha amylase. Now you know why am I using the word pancreatic alpha amylase. I hope you know the meaning now, right? It is also known as amylopsin. Amylopsin. So remaining 30% of the starch will be digested here in buccal cavity. Remaining 70% will be digested there in the small intestine. Okay, it will be digested there in the small intestine. Then bache, pancreatic lipase is also there, which will help in the digestion of your fat. And it is known as steapsin. Right, it is known as steapsin. Then you have, right, yes, then you have proteases as well. So when it comes to the proteases, again they are in inactive form. What are they? They are in inactive form. So bache, here we have 3 examples, right? We have the trypsinogen. When it is trypsinogen, gen word is there, means you are talking about the inactive form. Clear? Then you have chymotrypsinogen, right? You have the chymotrypsinogen, it is also inactive. Then you have the procarboxypeptidases. It's procarboxypeptidases. Clear? So trypsinogen, chymotrypsinogen, procarboxypeptidases, they are inactive, right? They are inactive. They are protein digestive enzymes. Clear bache? So trypsinogen, chymotrypsinogen, procarboxypeptidases is there, right? And other than that, one more, it is also protein digesting enzymes. Elastases is there, right? Elastases is there. And then bache, enzymes for the digestion of your nucleic acid. Nucleases are also there. Nuclease means which will digest nucleic acids. Like here you have DNAs, you have RNAs. So why do we say that pancreatic juices are the complete digestive juices? Why do we say that pancreatic juices are the complete digestive juices? Because they have enzymes for the digestion of all type of substrate. So pancreatic alpha amylase, amylopsin, lipase, steapsin, elastases, proteases mein they are inactive. So trypsinogen, chymotrypsinogen, procarboxypeptidase is the example. When it comes to the nucleases, DNAs and RNAs are there. So I hope it is clear to you all. Right? Right? Anji, clear? But say it is related to 11th biology, right? It is the chapter there in 11th biology. Open up your old NCERT, you will find it. So if there is any doubt, please let me know. So this is about the composition of the pancreatic juices. So pancreatic juices may only inorganic salts are there. What type of salts will be there? Only inorganic salts will be there. Clear, bache? Only inorganic salts will be there. Done? So now let's talk about the duct tree. Okay, oh wow, even this is nice. Okay, chalo. Let's talk about the duct tree now. So we have right lobe of liver. We have left lobe of liver. है ना? So from this right lobe and from this left lobe, this is my duct tree. Okay, from this right lobe, it is your right hepatic duct, bache. It is your right hepatic duct. This one will be your left hepatic duct. Clear? Here you have gallbladder. What do you have here, bache? You have gallbladder. So now the duct of gallbladder, it is known as cystic duct. Done? The duct of gallbladder is known as cystic duct. Isn't it? Isn't it? The duct of gallbladder is known as cystic duct. Clear? So now your cystic duct and your, right, see, right hepatic duct, left hepatic duct, they will join. See this part, right and left duct, they are joined, they are forming common hepatic duct. What are they forming, bache? They are forming common hepatic duct, isn't it? Now this common hepatic duct is joining with the cystic duct, right? This common hepatic duct, it is joining with the cystic duct, and when both of them they will join, they are forming, bache, bile duct, right? They are forming a common bile duct. Clear? I'm repeating this portion again. Right lobe of liver having right hepatic duct, left lobe of liver having left hepatic duct. Gallbladder is having cystic duct and common hepatic duct, they are joining, you are getting common bile duct. Now we have the pancreas also. It's like, let's say you are going to the market, right? You are going to the market, your mother is saying, okay, bring me this, bring me that. Your neighbors are saying, Arey, you are going to market, do this thing for me, and like this. Clear? Clear? So next to it, you know that we have the pancreas as well. What we have? We have the pancreas as well. So pancreas mein your main pancreatic duct, right? It is your main pancreatic duct. And I told you, bache, your main pancreatic duct is also known as duct of Wirsung, right? So now your main pancreatic duct and your common bile duct, they will join. Your main pancreatic duct and your common bile duct, they will join. So now when they will join, they will form what? Obviously liver and pancreas together. It's hepatopancreatic duct. What is it? It's hepatopancreatic duct. Clear? It's hepatopancreatic duct. So now here it is having the secretion of pancreatic juices are also there, and the bile is also there. Now this hepatopancreatic duct, na bache, it is forming this swollen portion. So here we call it as hepatopancreatic ampulla. What is it? It's hepatopancreatic ampulla, and it is also known as Waters ampulla. It is also known as Waters ampulla. Right? And finally it will open up here in the duodenum, in the small intestine, in the duodenum. So it is looking like a tree, so that's why it's my duct tree. Clear? It is looking like a tree, so that's why it is my duct tree. And here, bache, you people will see, before opening here, right, this hepatopancreatic ampulla is having the sphincter of Oddi. Right, here it is a sphincter. This opening is guarded by a sphincter, that is your sphincter of Oddi. Right, this is your sphincter of Oddi. Right, this is your sphincter of Oddi. Clear? And even your common bile duct, before joining here with this pancreatic duct, it is also having a sphincter, bache. Right, it is also guided. This is also guarded by a sphincter, that is your sphincter of Boyden. So that's important. Clear? So sphincter of Boyden is guarding your common bile duct before it is joining with the pancreatic duct. Done? So this is the duct tree. Shiva Kumar, if you are not understanding anything, please do not waste your time. It is a class 11 chapter, digestion and absorption. Right now we are talking about the duct tree. So don't waste your time here, please. Fine. So is it clear, bache? Right? So right and left lobe separated by falciform ligament. Just below the right lobe of liver, the gallbladder is present. Gallbladder is going to store and concentrate the bile. Gallbladder duct is cystic duct, so it will join with the common hepatic duct, and when cystic duct and common hepatic duct will join, they will form common bile duct. Now when this common bile duct it is guarded by a sphincter that is known as sphincter of Boyden, right? And then this common bile duct will also join with main pancreatic duct. As I said, main pancreatic duct is known as duct of Wirsung, and pancreas is also having one accessory duct, and that duct will directly open up into the small intestine, and this duct is your duct of Santorini, right? So it is the duct of the accessory pancreatic duct which will directly open in the small intestine. Okay, okay, right. So when they will join, so you will have hepatopancreatic duct, hepatopancreatic ampulla, and you know that this duct is guarded by a sphincter of Oddi, and finally it will open up in the small intestine, that is duodenum. So it looks like a duct tree, isn't it? It looks like a duct tree. Clear? Clear. So that's how all the secretions will finally pass to the small intestine, and that's how further, right, the digestion and maximum absorption will be there in your small intestine. Now see, you guys can understand this diagram. See the gallbladder, the cystic duct joining with hepatic duct, common bile duct. It is receiving your pancreatic duct, hepatopancreatic ampulla, finally open up here in the small intestine. Any doubt? See, any doubt? Done, bache? And look at the exocrine part of your pancreas. So it is having lobules. Fine, it is having these lobules. Done. Acini they are making your digestive juices. Clear, bache? Clear, bache? They are making your digestive juices. Done. So this is the diagram from NCERT. So I hope it's clear now, because next part that we need to start is the digestion of food, the physiology of digestion we need to start. Okay. Done. Next part is the physiology of digestion that we need to start. Fine. So that's important. Okay. Done. Okay, so just look at these diagrams. So now you know this duodenum will further join with your, duodenum, jejunum, ileum will further join with your large intestine, right? And you can see the internal structure of the villi we have already discussed. So just look at the cells, bache, tall and slender cells having microvilli. So that's why it is what? It is the brush bordered epithelium. Okay, so in small intestine, this epithelium is absorptive as well, secretory as well. Absorptive as well, secretory as well. Clear? Okay. See this beautiful diagram. Fine. This is that beautiful diagram. And next to it is this large intestine. So here you can see the cecum, and this one is the vermiform appendix, which is vestigial. We have already covered this part. So here, when cecum and ileum, they are joining, this is the ileocecal sphincter. So your ascending colon, transverse, descending, sigmoid. Finally, it will open up in rectum, and rectum is the place where there will be the storage of the fecal matter. So rectum will leads into anal canal, and then there will be the anus. Okay, then there will be the anus. So just look at this. This opening is anus. This is your anal canal. So anal canal is having external or extrinsic sphincter, internal sphincter. Done. So that's what we have covered. So look at these diagrams. Again, very beautiful they are. Right. So see this diagram, how beautifully it is, it is showing these villi and the crypts of Leberkuhns. Right. You can see the crypts here. And we see. Done? Any doubt? Sure. Okay. So next topic that we need to discuss is digestion of food. Right. So now it's the physiology of digestion, and finally then there will be this calorific value, and finally absorption, and the disorders will be there. So let's finish it today itself. Hai na bache? Let's finish the digestion of food first. Let's finish the digestion of food first. So if there is any doubt, firstly tell me. If there is any doubt, do let me know. Sure? So you know that, right? Firstly, there will be the swallowing of the food. Food will be swallowed, right? It is also known as deglutition. Swallowing is also known as deglutition. In the buccal cavity, there will be the mastication of food, right? There will be the mastication of food, right? The saliva will get mixed, the bolus will be formed. So that bolus will be swallowed, bache, and then it will be passed to the stomach. And you know that how will it be passed? It will be passed with the help of peristalsis. So peristalsis is rhythmic contraction and relaxation. I have explained it so many times, isn't it? I have explained it so many times. And then finally from the stomach may acidic secretions will get mixed. Now it is the chyme. So it is the chyme which will pass to the small intestine. Okay, so in small intestine, all that secretion, all that food, here it is making chyle. So it is the chyle which will be passed to the large intestine. That's how it works, right? It will be passed to the large intestine, right? So you know that when it is the digestion, it is the mechanical and chemical digestion, right? The churning of food, mechanical. Enzymatic action, it is the chemical digestion. So you know that buccal cavity will perform two main functions: mastication and facilitation of the swallow. These are the two main functions of the buccal cavity: mastication of the food and then facilitation of the swallowing of the food. So teeth and tongue will help in the with the help of the teeth and tongue, saliva masticate and mix up food thoroughly, right? So mucus in saliva helps in lubricating it, right? It will adhere the masticated food, it will, you know, join that masticated food, it will form the bolus. So as I said, bolus will be conveyed into pharynx and then into esophagus by this deglutition. So swallow the secretions, right? Now you know everything related to the digestive juices. So first of all, let's start with the, first of all, let's start with the digestion of carbohydrates. So bache, digestion of carbohydrate, it starts in buccal cavity. Clear? It starts in, yes everyone, it starts in buccal cavity. So first of all, in buccal cavity, 30% of the starch will be hydrolyzed. 30% of starch is hydrolyzed here. And do you know that which enzyme is present? So see, starch, salivary amylase, right, also known as ptyalin. You know that pH is 6.8 here, right? pH is 6.8 here. So what is going to happen, bache? This starch will form your maltose, your alpha dextrins. Am I right? It is going to form your maltose and the alpha dextrins. Yes or no? Yes or no? So this is going to form your maltose and alpha dextrins. Now, bache, the question here is, ma'am, what is the meaning of these maltose and alpha dextrin? Now, 30% of the starch will be hydrolyzed, right? 30% of the starch will be hydrolyzed. So you know that this maltose is a disaccharide. This is some of the starch will form the disaccharides and some will form dextrins. Dextrins are also having high molecular weight, right? They are also having high molecular weight, but not that as much as starch. Clear, bache? Not as much as starch. Now, after this, you know that stomach may no digestion of carbohydrates. Am I right? Stomach may there will be no digestion of carbohydrates because conditions are not optimum. We do not have any amylase here which can work under acidic conditions. Clear? So no digestion of carbohydrate will be there in the stomach. Is it done? Yes, bache. So next is the digestion of carbohydrate in small intestine. Next part is what? Digestion of carbohydrate in small intestine. Tell me if there is any doubt. Right, that's why, bache, firstly we have covered the biomolecule chapter. If you know biomolecule, then you know these terms, maltose, dextrins. So digestion of carbohydrates in small intestine. Now you know small intestine is going to receive the secretions of pancreas as well. It is having the intestinal juices as well. It is having the bile as well. So first of all, there will be the action of pancreatic juices, right? What will be there, bache? There will be the action of pancreatic juices. Right. So now, can you tell me in pancreatic juices which amylases are there? Yes. Can you just tell me which amylases are there in the pancreatic juices? Can you just tell me which amylases are there in the pancreatic juices? You know the answer? You know the answer? Of course, bache, it's amylopsin. Right, and it's a very important enzyme because 70% of the starch will be digested here. So it is the amylopsin, pancreatic amylase, right? And again, this enzyme also needs chloride ions, right? So starch, it will form your maltose plus your alpha dextrins here, right? So 70% of the starch will be hydrolyzed. What will be done, bache? 70% of the starch will be hydrolyzed here. Are you getting it? It will be hydrolyzed here. Done? Done? So in the small intestine, firstly pancreatic juices are going to act on this carbohydrate. Then there will be the action of intestinal juices, or you can say that succus entericus. Right, then there will be the action of succus entericus. So now what is going to happen? There will be the further breakdown of these alpha dextrins. Clear? There will be the further breakdown of these alpha dextrin. Right. So see, first of all, your maltose, it's a disaccharide. But when I explain it in the in the biomolecules, now I always used to say that these three disaccharides are very important for you, right? Always remember that these three disaccharides are made up of which monosaccharide. So when it comes to the maltose, always remember it will give you 2 glucose, right? So which enzyme is going to act on it? It is your maltase. So in intestinal juices, I told you now, we have the disaccharidases, we have the enzymes that can break the disaccharides into the monosaccharides. So maltase, then comes your sucrase, then comes your sucrose. Sucrose is the invert sugar. So your invertase and sucrase can act on it, bache, and you will get glucose plus fructose. You will get glucose plus fructose. And then we have the lactose as well. And when it comes to the lactose, lactase is going to act on it. You will get glucose and galactose. Clear? You will get glucose and galactose. Fine. Fine. So we have the dextrases as well, which will break these dextrins. Okay. We have the dextrinase as well, which will break this dextrin. Let me write it. Done. So that's how it will act. So finally, you will get the simple form, isn't it? Finally, you guys are going to get the simple form. Let me write it here. Okay. See, dextrin. It is also having dextrinase. And finally, you will get the glucose. So this is the carbohydrate digestion. Please take the screenshot as well. Okay, please take the screenshot as well. So this enzyme is very important. This pancreatic amylase is also known as amylopsin. Okay, it is also known as amylopsin. Sure? Done, bache? Done? Sure? Any doubt? Okay. So next is your digestion of proteins. Right? Just in NCERT it is mixed, but we will discuss it separately. So next is your digestion of proteins. What is it? It is the digestion of proteins. Clear bachche? So let's start with the buccal cavity first. Now you know that in the buccal cavity we do not have any protein digesting enzyme, isn't it? We do not have any protein digesting enzyme, but in the buccal cavity, bache, the denaturation of protein will occur. Like if we are eating any uncooked protein, so that will be denatured. Just say raw milk, raw egg, right? Milk, right? So it will be denatured there. Okay, what is going to happen there? Denatured here. Are you getting it? Are you getting it? So saliva can denature uncooked protein. Right? Now you know not denature, to convert it into one form, like tertiary will become secondary, secondary will become primary. Right? Right? Tertiary will become secondary, secondary will become primary. So uncooked proteins will be denatured with the help of saliva, right? Jaise you will see it in the case of raw milk, raw egg, unboiled milk. So they have done. So saliva can denature uncooked protein, like protein present in raw egg, unboiled milk, etc. But there will be no digestion of protein in the saliva, in the buccal cavity, because there is no peptidases, right? There is no peptidases. Is that clear? Is that clear? Yes? Okay. So saliva can denature, but do not have proteases. Done. Now next is what is going to happen in stomach, right? What is going to happen in stomach? But before discussing this part, let me tell you, bache, in nature the protein digesting enzymes, they are secreted in inactive forms. They are zymosins basically. They are the proenzymes basically, because if they will be in activated form, they can even digest the extracellular matrix protein. That will not be good, isn't it? That will not be good. So when you are talking about the protein digesting enzymes, they are released in the protein digesting enzymes. They are released in an inactive form. They are released in an inactive form. So for that inactive form, we use the word proenzyme, or you can also call it as zymosins, right? You can also call it as zymosins. Are you getting it? These are the proenzymes, or you can also call it as zymosins. Is it clear? Yes, bache, is it clear? Yes or no? Yes or no? Yes or no? Right? So these peptidases, these protein digesting enzymes, these are the peptidases. Now I told you why they are released in inactive form, because otherwise they will digest the cellular protein. These peptidases are also of two types. Like we have exopeptidases, we have endopeptidases. Do you know that? They are of two types, exopeptidases, endopeptidases. So exo means outside, right? So they will break peptide bonds of, right? Break peptide bonds of terminal. Exo means outside. So it will break peptide bonds of terminal amino acids. Clear, bache? And when it is the endopeptidases, right? So they will break peptide bonds of non-terminal amino acids. Right, let me explain this part. I'll quote one example. Like here, you are talking about the enzymes which are there in your intestinal juices, like your aminopeptidases are there. Here, you are talking about the enzymes present in digestive juices like pepsin, trypsin. See, it is in gastric juice, it is in pancreatic juice, right? So first of all, this is the point. Now, bache, when you talk about the proteins, you know that proteins are made up of amino acid, and in one protein, it is not mandatory that you will be having only one chain, right? It's not mandatory that you will be having only one chain. It's not mandatory, okay? That protein can be made up of many polypeptide chains. Okay, it can be made up of many polypeptide chains. So when we are using exopeptides, so they will break these terminal, right? They will break these terminal amino acid. They will break the peptide bonds present here. It is the peptide bond which is linking two different amino acids. So if I'm using endopeptides, they will break the peptide bond, right, of non-terminal amino acids. Clear, bache? Clear, bache, like your pepsin and trypsin. Okay, like your pepsin and trypsin. So basically such enzymes are going to break your high molecular proteins into the small, right, into the proteins having low molecular weight, like peptones will be formed. Okay, so that's what we need to discuss. So first of all, tell me this part clear? This part clear? Now, in the stomach, what is happening? Right, in the stomach, what is happening in the stomach? You know that we have the HCl. HCl, it is secreted by parietal cell, right? We have the HCl which is secreted by parietal cell, isn't it? Isn't it also known as oxyntic cell? So now this HCl, like in the stomach, but this is what you need to remember, that which proenzymes are present in stomach? Pepsinogen. So pepsinogen will become pepsin due to the presence of this HCl, right? Your pepsinogen will become pepsin. Now this pepsin is having, right, this pepsin is the active form, and bache, this pepsin is having the autocatalytic property, right? What is the meaning of autocatalytic property? Can you tell me? So this pepsin is going to show autocatalysis. Okay, so what is the meaning of autocatalysis, right? Like in the digestive juices, in the gastric juices rather, we have the propepsin or we have the pepsinogen. Now HCl is going to activate it. Okay, when HCl will be released, it is going to activate it. So pepsinogen is going to form the pepsin. Now this pepsin is like, okay, I need another pepsinogens as well. Let me activate them as well. So this pepsin will, when once it is activated, it will active another inactive enzymes, right? It is going to active another inactive enzymes. Clear, bache? Clear, bache? Right? So it will form another pepsins. Okay, so this is the autocatalysis. It is helping in its own activation once it is activated. Clear, bache? Are you getting it? So pepsinogen will form pepsin. Now this pepsin will act on proteins having high molecular weight. This pepsin will act on proteins, and you know that proteins are having high molecular weight. Yes or no? Proteins are having high molecular weight. Yes or no? So it will form proteoses plus your peptones. So proteoses and peptones are nothing. They have the low molecular weight in comparison to these proteins. Clear, bache? In comparison to these proteins. Clear? Clear? So this is about the role of the pepsin. Now, I told you that prorenin is also there. But this prorenin is found in the infant stomach. Okay? In adults, you are not going to find this enzyme. It will be there in the adult. It will be there in the infants only. Right? So now, what is the role of this prorenin? First of all, this prorenin, of course, in the presence of HCl, it will be activated. It will be activated. It will form the renin. What will it do? It will form the renin. What will it do? It will form the renin. Now, actually, this renin is acting on the milk protein. Okay, but in the milk, there is a sugar as well. There is a protein as well. Milk sugar is your lactose. Do not ever confuse it. Milk sugar is lactose advat. Milk advait. Okay. Advait. So milk sugar is lactose and milk protein is casein. Milk protein is what? Casein. So this renin is going to act on casein. Fine. This renin is going to act on casein and it will form para-caseinate. So actually this renin helps in the digestion of milk. Now, as I said, it is present in the case of infant stomach, means in our case, we do not have this prorenin. So the point is how milk will be digested in our body. Actually, our system is designed in this way. We are the mutated forms, okay? We are the mutated forms. We are taking the milk. So our body has adapted accordingly and digesting it. But you know that many people, right? Many people, they cannot digest the milk after an age. Why? Why? Because this prorenin is absent. Now in our case, what is happening? We do not have this renin, okay? But the pancreatic protein digesting enzymes, they can help in the digestion of that milk protein. So for our body to digest milk, right? For our body, it is quite difficult to digest the milk, but still we used to take it. Right now, I'm not telling it in the context of being lactose intolerance. Right now, I'm telling you about the digestion of the milk protein, casein. Lactase is absent, so lactose will not be digested. So lactose is the milk sugar. Milk is having that sugar. So some people, they do not contain the lactase. So they do not have the lactase. They cannot digest the milk sugar, lactose. So in their body, if they are taking milk, that lactose will be there in their system only because it is not degraded, right? So obviously it will get, the bacteria will start feeding on it. And then obviously the stomach will get disturbed. All that gas, turbulence will be created in the stomach. Right, so we are basically the mutated form because we are taking the milk. Like if I talk about myself, I like to drink milk. So I used to have that, right? So my body is putting extra efforts to digest it. So we are kind of, we are mutated, right? Because we are giving our body that particular thing. So our body is like, okay, I have to do something for its digestion. So that's how it works, right? So basically milk is advised for the infants only, right? They need it more, not in our case. So in our case, we take it, so our pancreatic enzymes are working on it for its digestion. Fine, fine. So that's the science behind. So now casein, renin will act, you will get para-casein, right? You will get para-casein. Now on this para-casein, right? How this para-casein will be digested? Now this para-casein will join with the calcium ions and it will form calcium para-caseinate. What will it do? It will form calcium para-caseinate, right? So it is actually that curd. This is babies. They pass that, they regurgitate that now. That is your calcium para-caseinate. What is it? It is your calcium para-caseinate, right? So finally, bache, on this calcium para-caseinate, pepsin is going to act. And then again, it will form peptones or proteoses. So that is the digestion of proteins in the case of stomach. So in the case of stomach, that's how it will work. Done? That's how it will work. Done? So when we will become adults, so in our case, the milk digestion, the function will be done by pepsin, and then pancreatic proteases are going to help here. So this is what you need to remember. Fine. This is what you need to remember. Okay. So protein digestion, you know that it will start in the stomach, right? It will start in the stomach. So that's how it will be done in the stomach. So next part is your intestine. So intestine, may you know that again the further proteins will be digested here in intestine. So in intestine, first of all, there will be the action of pancreatic juices. So now you know that even pancreatic juices, they contain inactive proteases. Isn't it? Now even the pancreatic juices, yes bache, now even the pancreatic juices, they are also having the protease, isn't it? They also contain inactive protease. They are also present in pancreatic juices, right? Right. Now, bache, it's very interesting. Now you know that in the small intestine, you have everything, right? Small intestine is also having its own intestinal juices, right? That is succus entericus. Now, here in the small intestine is also receiving pancreatic juices, right? Small intestine is also receiving the bile. So ultimately in small intestine, everything is present. Now, if you remember, right? If you remember, I told you already that in intestinal juices, that is intestinal juices means succus entericus, we have many enzymes, and I gave you the example of one, that is enterokinase. Remember? Remember? In today's lecture also, in today's lecture I told you about it. So what we have? We have the enterokinase there. What we have there? We have the enterokinase there, which I told you it's a non-digestive enzyme. Now, what is the meaning of being non-digestive enzyme? Like this enzyme is not going to digest any respiratory substrate. Then how is it helping? Let me tell you, right? See, we have trypsinogen in pancreatic juices, right? We have trypsinogen in pancreatic juices. That trypsinogen will form trypsin, which is a endopeptidase. So how the trypsinogen will become trypsin? Trypsinogen means inactive form. Trypsin means active form. So how is it possible? Of course, here there will be the role of this enterokinase. That's very important part, students. Right? This reaction, this part is very, very important. Please note down. Right? This part is what? This part is very, very, very important. Okay? Enterokinase. Fine, this enterokinase is helping in the digestion here. Done? Any doubt? Right? Any doubt? Sure. So ultimately this enterokinase, which is the, okay, and here calcium ions are also required. So trypsin. Now trypsin is also having the autocatalytic property. Clear? This trypsin is also having the autocatalytic property. So once it will get activated, it will active another trypsin molecules. And moreover, bache, another inactive enzymes like your chymotrypsinogen, okay, your procarboxypeptidases, they all will be activated in the same way. Clear? They all are going to get activated in the same way. Okay? Okay. So here your trypsin will help. Done, bache? Here your trypsin will help in their activation. So that's how it is. It works. So if intestinal juices and pancreatic juices are not present simultaneously, pancreatic proteases will not get activated. There will be no digestion of the proteins then. Clear? There will be no digestion of the proteins then. So chymotrypsin will form, chymotrypsinogen will form chymotrypsin. Procarboxypeptidase will form carboxypeptidase. Any doubt? Any doubt? And further, all of them, right? Now they are activated. So your trypsin, your chymotrypsin, your carboxypeptidase, they will act on proteins, right? And what will they do? They will help in their further digestion, right? So proteins, even the peptones, even the peptides, okay, proteoses. Proteoses. So all of them, right? The proteins, the peptones, and the peptones and the proteoses, right? They will be digested by these enzymes, and finally you will get dipeptides. Now you know na dipeptides having 2 amino acids. It will form dipeptides. Clear? So that's how your pancreatic juice is going to act on these proteins. Clear? So this step is very important, bache. Question can come from this part. Question can come from this part. Okay? Okay. So finally there will be the action of, intestinal juice. right? So intestinal juice mein you know that dipeptidases is there. So finally dipeptides, on it dipeptidase will act and you will get amino acids, the simpler form. Fine, it is the simpler form. Clear bache? Finally the amino acids will be there. Okay, sure, sure bache? Any doubt? Any doubt? So that's how the proteins will be digested in the stomach. So next is the fat digestion and the fat absorption. So see, so bache, all the reactions are from NCERT, so it will be better to revise it. Okay, so see, these reactions are given here, right? So in the NCERT, it's all mixed. Like they are discussing it in this way, that what is happening in buccal cavity, what is happening in stomach. But I'm telling you these things individually. Like in the case of carbohydrate, that's how it will work. In the proteins, that's how it will work. So this is how we are discussing it basically, right, bache? So, right, we have covered it up to this part. So now we have to start with the fats. Clear? We have to start with the fats, and fats may digestion of fats and even the absorption of fats we will discuss all together. Clear? We will discuss all together, and I have added this flow chart as well. So I'll share the PDF with you people, and then you can, yes, you can revise it from that particular PDF. Okay. So I think we need another 1.5 hours to finish this chapter. So I think today in one go we should finish it. What say, bacho? है ना? We can finish it in one go. What say? We need another 1 hour or 1.5 hours. Because the digestion of fat and the absorption of fat is the main topic. Nucleic acid digestion is very easy, and then your calorific value and the nutrition value given in NCERT, again a very easy topic. Okay, and then comes the disorders. So disorders may, there is nothing. Okay, so bache, now it's 9:15, 9:12 actually, take it as 9:15. We'll start the class at 9:30, right? Again you have the break time, you can have your dinner. Even I'll have my dinner. Then from 9:30 to 11 we'll finish it. Fine. From 9:30 to 11 we will finish it. Clear? Clear, clear, clear. So just have your dinner. Hi, hello everyone. So can we start? So done with your dinner and everything? Are you feeling energetic? So now we will continue. We will continue with the digestion of fat and then absorption and finally the disorders, right? So disorders particularly we are going to just we will follow the NCERT for that only, right? And for fat digestion and absorption, it's a very simple topic. We will finish it in one go. Okay. So now let's start the digestion of fats, which is not so easy. Students, do you know that fats used to provide us more energy in comparison to the carbohydrates? Right? When we talk about the fats, they provide us more energy in comparison to the carbohydrates, but still our main respiratory substrate is carbohydrate. Why is it so? Because for the oxidation of the carbohydrates, it's quite easy, but for the oxidation of fats, we need a lot of energy, and the fat digestion, it takes time. Okay, and in our body, we need some immediate source of glucose when required. That is why. Okay, that is why. So now when you talk about the digestion of fats, let's start with the mouth, right? Let's start with the buccal cavity. So you know that buccal cavity may we do not have any amylase, isn't it? We do not have any lipase, isn't it? Yes or no? Now you are going to answer the question. So you know that saliva, it doesn't contain, right? In the buccal cavity, there will be no digestion of fat because saliva, it doesn't contain lipase. Yes or no? There is no lipase there in the saliva. That's why. Now, when you talk about the stomach, you know that gastric lipase is there, right? I'm not denying that gastric lipase is not present there. Gastric lipase is there, but here the digestion of fat is negligible. Only 1% of the fat will be digested. Okay, only 1% of the fat will be digested. So again, it is the negligible amount of digestion. Clear? Now, moreover, when you talk about the fats, bache, what you discuss, right? I hope you all have attended the lecture of the biomolecules. So when you talk about the fats, fats contain your glycerol plus fatty acids. Clear? Your glycerols plus fatty acids, they are joined by ester bond and you will get the fats like your 1 glycerol plus 3 fatty acids, they will form triglycerides, right bache? They will form triglycerides, the most common type of fat. When there is glycerol plus 2 fatty acids, they will form diglycerides. When there is glycerol plus 1 fatty acid, it will form monoglyceride. So ultimately in fat digestion, we are going to break these triglycerides into di, then into mono, and further we will break that monoglycerides into glycerols plus fatty acids. Okay, we are further going to break them into glycerol plus fatty acids. So that is how the digestion of fat will take place. Clear, bache? That is how the digestion of fat will take place, right? So in the gastric lipase is there in stomach, right? But here again the negligible amount of digestion will be there. So now mainly it will start in intestine. Fat digestion will mainly start in intestine, and first of all there will be the action of bile. What is going to happen first, bache? There will be the action of bile first, right? Now you know bile is formed by liver. It is meant for the it is helpful for the emulsification of fat. Clear, bache? So first point is, what is the meaning of emulsification of the fat? Let's discuss it here. Okay, so for the fat digestion, the first thing that we need to discuss is action of bile. What is it? It is the action of bile, right? Action of bile. So first of all, the fat, it will be emulsified with the help of bile salts, right? What bile salts will do? They will emulsify fat. Now, what is the meaning of emulsification? Emulsification means the large fat droplets, right? The large fat droplets, they will be covered with the bile salts, and bile salts will break it into small fat globules. Are you getting it? Right? So what is the meaning of emulsified fat? Yeah, what is the meaning of emulsification of fat? That large fat globule, there will be the breakdown of large fat globule into the smaller ones. Right. So what is going to help here? The bile salts are going to help here. Right, the bile salts are going to help here. That's the first point. This is the emulsification. So bile salts, they are covering this fat globule and they are breaking it down. So this is the very first step here. Clear, bache? This is the very first step here. So your salts are going to help here. Now you know about that salts. Sodium bicarbonate is there, sodium taurocholate is there, sodium glycocholate is there. Clear, bache? Sodium glycocholate is there. Now the next step is this emulsified fat. What is going to happen to this emulsified fat? Bache, to emulsified fat, right, now there will be the action of pancreatic juices. Fine, there will be the action of pancreatic lipase, and you know the name of that pancreatic lipase, that is your stepsin. Right, so stepsin is going to act on these small fat droplets. Are you getting it? Stepsin is going to act on this small fat droplet. So it is going to break it, right? It is going to break it. So it will break it into fatty acid plus diglycerides, right? Fatty acid plus diglyceride. Bache, again I'm repeating this point. It is the triglyceride, which is a very common form of fat, right? So here I told you already that glycerol, which is your trihydroxypropane. Glycerol, which is your trihydroxypropane, right? This glycerol will join with fatty acid with the ester bond. So if one glycerol, because it is having 3 OH group, so if one glycerol is joining with 3 fatty acid, it is forming the triglyceride. If it will join with 2 fatty acids, di. If it is joining with 1 fatty acid, it is the monoglyceride. So ultimately here the emulsified fat, right, that stepsin is going to act on it, and it is the main lipase, remember this. Okay, it is the main lipase. So you will get fatty acid plus diglyceride. Then again, bache, what is going to happen? Can you tell me? On this diglycerides again, your lipases will act. Right, on this diglyceride again your stepsin will act, and then again it will break it down into fatty acid plus monoglyceride, right? Fatty acid plus monoglyceride. Now bache, the role of intestinal enzymes will also be there. Okay, so that intestinal enzymes are also going to act on these diglycerides. You know, na, intestinal lipases are also there. Intestinal lipases are also there. So it will again break it into fatty acid plus monoglycerides. Clear? And then again monoglycerides. On monoglycerides, your intestinal lipases will act. Okay, on these monoglycerides, intestinal lipases, they will act, and you will get glycerol plus fatty acids, the simplest form of the fat here. Clear, bache? The simplest form of the fat here. Is that clear? Is that clear? Yes or no? So that is how the digestion of the fats will be there. So to understand the physiology of digestion, you should know that carbohydrates, like your starch, it is made up of what? Your proteins, your nucleic acids, they are made up of what? Then the topic is going to be very easy for you. And let me tell you, bache, bile salts are very important. They are even helping the activation. Right, bile salts, they activate lipases, right? They activate lipases. Clear, bache? This is also a very important role of the bile salts as well. Clear? Clear? Right. So now, bache, with this part, right, let's discuss the absorption of fat as well, because absorption of fat is not that easy. Like in the case of carbohydrates, we are clearly saying that, they will be converted into simpler forms, like your glucose, like your fructose. So easily they will be absorbed by the cells, or might be. ATP will also be used, but very easily it will be done. But when it comes to the fat absorption, it is different. You know that fats are insoluble in water. Right? You know that fats are insoluble in water. So obviously they will not get easily absorbed, right, in our body. We need the help of lymph vessels. We need the help of lectins. That is what we will discuss here. Okay. Okay, so absorption of fat. You tell me what you know about this topic. Absorption of fat. What do you know about it? What do you know about the absorption of fat? Tell me. See, you know that, as I said, glycerols and fatty acids, they are water insoluble, isn't it? They are water insoluble. So what will be the first step here, right? These glycerols and fatty acids, they are going to form some structure. They will form the measles, right? What will they do? They will form the measles. Are you getting my point? Right? These droplets, right? These fatty acids and glycerols, they will first incorporate it into small droplets, and we will call it as measles. Just a minute. Glycerols and fatty acids are incorporated into small droplets, right? They are incorporated into small droplets, right? They are incorporated into glycerols and fatty acids. They are incorporated into small droplets, which are measles. But these measles are water soluble, let me tell you, right? These measles. First of all, now you know that in the intestine, we have the intestinal cells. These enteroendocrine cells are there. These intestinal cells are there. So what is going to happen? These measles will be, right? These measles will be absorbed by the intestinal cells. This is what you need to remember, right? The measles, they can move into this intestinal mucosa. Measles, this is as such NCERT bache. These measles are, yes, these measles can enter in the intestinal mucosa. Right, they can enter in the intestinal mucosa. Now these droplets, they will enter here in intestinal mucosa. That's not the only part. No, here these measles will be reformed. You know, again they will form chylomicrons. Like you can say that they will form globules which are coated with the protein. And let me tell you, that part is water soluble. Even measles are water soluble. Measles are, just keep one thing in your mind, that measles can be absorbed by this intestinal mucosa. Okay, measles can be absorbed by the intestinal mucosa. That's what you need to remember. Clear, bache? Right, they can move into the intestinal mucosa. Okay, done. So now these measles here in intestinal mucosa, measles are reformed. What is going to happen here, bache? Measles are reformed into small fat globules which are coated with protein. They are reformed. They will form again, and they are reformed into small fat globules coated with protein. Clear? Coated with protein. And what is it? They are chylomicrons. Okay, they are chylomicrons. And yes, chylomicrons are water soluble. That's important. These chylomicrons are what, bache? These chylomicrons are water soluble. Clear, bache? Clear? So they will be transported into the lymph vessel. Okay, and then ultimately they will pass to the, right, these chylomicrons, they will be absorbed by lymph vessel, and that you know which lymph vessel is there, that is your lacteal. Okay, lacteals are going to absorb it, then from lacteal it will pass to the bloodstream. So that is how they are absorbed. Clear, bache? That is how they are absorbed. Are you getting it? That is how they are absorbed, and they will be utilized in the body. Clear? So obviously their absorption is also a very different. Their absorption is also different from the absorption of that amino acid and the monosaccharides. Done? So that's how you have to remember that. Firstly, they will form. Chalo, let's revise it quickly. So firstly, on fats, on large droplets, bile salts are going to act. They will emulsify it. They are going to break it into small droplets. Now these bile salts are also activating the lipases. Clear? So pancreatic lipase, which steps in, it is the most important enzyme. So that pancreatic lipase steps in, it will act on emulsified fat and it will form fatty acids and diglycerides, right? Or you can simply write here, it can even form triglycerides here. Then further, intestinal lipases will act on it and they will further break them, right? They will further break them. So that's how it works. That's how it works. So you should know that fats are made up of what. And again, I'm telling you, please check the biomolecule lecture then. Okay, do check the biomolecule lecture if you have any doubt in this part. And finally, for absorption, mesocyl formation will be there. These mesocyls will be reformed here in the cell. They will form protein-coated small fat globules, chylomicrons, and they will be absorbed by the lymph vessel lectile and finally pass into the bloodstream. So that's how it works. Clear, bache? That's how it works. So now, the next part is the digestion of your nucleic acid. Clear? Next part is the digestion of your nucleic acid. Now, bache, when you talk about the nucleic acids, again, biomolecules. Nucleic acids are made up of polynucleotides, right? It is basically a polynucleotide chain. Nucleic acids are what? They are basically the polynucleotide chains. And these nucleotides are what? Like so many nucleotides, they join, they form the nucleic acid. So nucleotide include your nitrogenous base, sugar plus phosphate. Right, it includes your nitrogenous base, sugar plus phosphate, and nitrogenous plus sugar is known as nucleoside. So it's nucleoside plus phosphate that will make nucleotide. So when it comes to the nucleic acid, we have two examples. We have DNA and we have RNA. Clear? We have DNA and RNA. So for the digestion of DNA, we have DNAs. For the digestion of RNA, we have RNAs. Right, so these are the nucleases. So nucleic acids will be digested with the help of enzyme nucleases. Right, they will be digested with the help of enzyme nucleases. So we have DNAs, we have RNAs. So DNAs, right, DNAs, it will act on, see here, you will get nucleotides and then nucleosides. Just look at it. Right, so DNAs, from DNA, it will form nucleotides, and then you know that, right, pancreatic juices are having nucleotidases that will form nucleosides, and then your intestinal juices, they have nucleosidases, right? They have nucleosidases. So finally, you will get that nitrogenous base plus sugar. So that's how it works. Clear, bache? That's how it works, right? That's how it works. So see, nucleotides, right? In intestine, nucleotidases, nucleosidase will form sugar plus bases. And di and monoglycerides, here you will get fatty acids plus glycerol. So that's how it is going to work. So this is all about the physiology of the digestion. Clear, bache? This is all about the physiology of the digestion. Okay. Okay, so next part we will discuss from the NCERT only, right? NCERT is more than sufficient to discuss the next portion. Okay, so I'm not going to write the point. So with the help of NCERT, we are going to clear it. Clear, bache? And even it will save our time. Already, we started the class at 5, hai na? So it's 9:52. So see, so no significant digestive activity occurs in the large intestine. So actually, bache, in the large intestine, here is the absorption of water, minerals, and certain drugs. Certain medicines, they will be absorbed in the large intestine. Clear? They will be absorbed in the large intestine. And moreover, secretion of mucus will be there in large intestine. Why? So that undigested food will be adhered. It will be joined together. Clear, bache? To form the feces. So secretion of mucus, which will help in adhering the waste particles together and lubricating it for the easy passage. Done? Done? So undigested, unabsorbed substance, you know that this is the feces, right? So it is defecation, right? So when we pass undigested food, that is feces from our body, we call it as defecation. Right? It is known as defecation. Clear? So, bache, defecation and excretion. You know these two terms are different. Defecation and excretion. Do you know these two terms are different? See, in defecation, there is the, right, undigested food will be passed out from our body. Undigested food will be passed out from our body. And in excretion, bache, it is the metabolic waste, right? After all the chemical reaction, that nitrogenous waste, that metabolic waste, which is the result of chemical reactions, it will pass out. That is excretion. But when it is defecation, in defecation, the food is the undigested food. So if it is undigested, means it has never entered our system, right? It is not taking, it has done nothing, it has to do nothing with our metabolism. Clear, bache? Clear? So this is the defecation and that is the excretion. So excretion means there will be the removal of the metabolic waste, there will be the removal of the nitrogenous waste. But here there will be the removal of the undigested waste. Clear, bache? So it enters into the cecum of large intestine through ileocecal valve. I told you, right? So ileocecal valve will prevent the backflow of this undigested food into the small intestine, right? So ultimately it will be temporarily stored in rectum till defecation. Okay, it will be stored in rectum till defecation. Done? Done, bache? So when you talk about the GI tract, about this gastrointestinal tract, bache, so neural regulation is also there and hormonal control is there. So I have given you the examples. Neural regulation, where we were talking about the parasympathetic and sympathetic fibers. I told you in the case of saliva as well, parasympathetic fibers will increase the secretion of saliva, sympathetic fibers will decrease it. Even I told you about the Meissner and Auerbach plexus also, right? So neural regulation, nerves are involved and hormones are also there, right? Hormones are also there. So first of all, the sight, smell and presence of food in the oral cavity. Even when you look at the food, why do our parents, our teachers say that when you are eating something, right, just focus on that food, right? Do not watch any movie or TV. And honestly speaking, even I watch, TV while eating the food. It's a bad habit. Literally, it's a bad habit. Why? Because all the senses are not involved in that, right? We are focused somewhere else. So if we will totally focus on our food, right, that smell, right, sight, see, you can read it here, the sight, the smell or the presence of food in oral cavity, it will stimulate our secretions, right? So if we will only focus on our food, our secretions, our digestive juices, their secretions will be induced, bache, and obviously that will be very helpful for the proper digestion. Okay, so gastric and intestinal secretions are also stimulated by neural signals, I told you, right? Even I told you about that parasympathetic and sympathetic fibers. So you know that the muscular activities of different parts of alimentary canal can be moderated by neural mechanism, right? Both local and through CNS. Like your central nervous system can directly control it, and local, that nerve fibers which are present in our alimentary canal, they take care of that, right? And even hormones are there. Like I have given you the example of gastrin. Even I told you about the secretin also, the first hormone discovered. And when I was telling you about the pancreatic juices, I told you pancreozymin, right? I told you about the pancreozymin. So pancreozymin will increase the secretions of that pancreatic juices. So hormonal control is also there. Clear, bache? Clear, bache? Hormonal control is also there. Done? Done? So that hormonal part, obviously, we will discuss later. We will discuss it in chemical control and coordination. Okay. So this part, see, this is important. Calorific value of protein, carbohydrate and fat. That's what we need to discuss. Here in this paragraph, it is from NCERT. We have two words. One is the calorific value, another is the physiological value. Let me quote the example here. Okay. One is the calorific value and another is the physiological value. Yes. Okay, one is calorific, one is physiological. I'll give you one very simple example. Just every day you make target table or timetable. You used to make target table or timetable. Right, as per your target table or timetable, today you are going to study for 10 hours. But you people study only for 7 hours. Right, again, that is also very good, huh? If you have decided that you are going to study for 10 hours and you are studying for 7 hours, again it is good, let me tell you. Now the point here is, these 10 hours, the one that you have decided, right, for you it's theoretical, practically it is not possible, for you it is theoretical. So these 10 hours, right? That will be the calorific value here, right? That will be the calorific value here. And 7 hours are the physiological value. Like it is same like something is, you know, theoretical and practically. You know that things are different theoretically and practically. So here in this paragraph, we talk about the different food items. We have carbohydrates, we have proteins, we have fats. They are our macronutrients, right? Carbohydrate, protein, fats, they are our macronutrients, isn't it? Isn't it, kids? Now, when it comes to the calorific value, it is a value, it is that energy that we can get. It is a kind of theoretical value that if you have that much gram of carbohydrate, so this is the amount of energy you should get, calorific value. But actually in our body, after all that metabolic reactions, the energy that it is giving, actual energy that it is giving is the physiological value. That's all. So you have to remember the values here. Why? Because it is given in our NCERT. So if something is given in our NCERT, it is the most important thing. है ना? So we cannot miss it, isn't it? We cannot miss it. Clear? So here you people can see the energy requirement of animals and energy content of food. Energy requirement of animals, energy content of food are expressed in terms of measures of heat energy. How do we measure the requirement, right? Our requirement and even the energy content of food in terms of heat energy, because heat is the ultimate form of all the energies. Okay, simple. Heat is the ultimate form of all the energy, so that is why we measure it in the form of heat energy. Fine. Now, this is often measured to as calorie or joule, which is amount of heat energy required to raise the temperature of 1 gram of water by 1 degree Celsius. Why you are studying physics and chemistry, I hope you can understand it in a better way. We are talking about the heat energies because it is the ultimate form of energy. We are measuring it as calories or the joules. Okay, CAL calories, joule J. Okay, so what is it? What is it? It is actually the amount of heat energy which is required. What is this calorie and joule if I have to define? That it is the amount of energy which is required to raise the temperature of 1 gram of water by 1 degree Celsius. I have 1 gram water. I need to raise its temperature. The amount of heat energy that I'm using is, that is how I will define this calorie and joule. Clear, bache? Clear, bache? So this value is the tiny amount of energy, right? So physiologists commonly use kilocalorie or kilojoule. So when I am defining calorie and joule, I am saying the amount of energy which can raise the temperature by 1 degree Celsius, right? 1 gram water and its temperature, we need to raise it by 1 degree Celsius, right? So it's a very less amount of energy. So which units are we using? We are using kilocalorie, we are using kilojoule. We are using kilocalorie, we are using kilojoule. Clear, bache? So 1 kilocalorie is the amount of energy required to raise the temperature of 1 kg. Now when it is kilocalorie, we are saying 1 kg of water by 1 degree Celsius, right? So nutritionists, they refer to kilocalorie as the calorie or joule, right? So the amount of heat liberated from complete combustion of 1 gram of food in a bomb calorimeter, right? It is the gross calorific energy value. So when I'm saying gross calorific energy value or gross energy value, so what is it? The amount of heat which will be released from 1 gram of food when we are doing its complete combustion, right? We have a closed metal chamber, right? We have a bomb calorimeter which is closed. It is filled with oxygen. So here you are going to, right, the complete combustion of 1 gram of food will be there. So the energy that will be released is calorific value. But in our system, it's different. Like let's say outside the body, 1 gram of carbohydrate is giving, in that bomb calorimeter, 1 gram of carbohydrate is giving a different amount of energy. But practically in my body, right, practically in my body, it is behaving in a different manner. Let's say I'm preparing you people for an interview. Okay, in front of me, you will give your best, but might be on the final interview day, maybe you will get nervous, or obviously the deviations will be there from whatever we have prepared. Clear? Same is the case here with this calorific value and the physiological value. So amount of heat which we will get in the bomb calorimeter, calorific, but the actual amount of energy combustion, the actual amount which will be there in our body is your physiological value. So these values you need to remember, right? Jaise gross calorific value of carbohydrate is 4.1 kilocalorie per gram. But if you talk about its physiological value, it's 4 kilocalorie per gram. Minor differences there, but difference is there. For proteins, it is 5.65 kilocalorie per gram. in bomb calorimeter, but in our body, see, it's 4 kilocalorie per gram. The difference is, right, the difference is there. And then, right, 9.45 calorie kilogram, right? And in the case of physiological value for the fats, it is 9. So that's what you need to remember. It is important. Clear? The question can come. Suppose karo here, let's say in your food item, 10 gram of carbohydrate is present. So they can ask you what will be the calorific value or what will be the physiological value. So if they are asking the calorific value, so 10 into 4.1 you have to do. And if they are asking the physiological, 10 into 4. That's how you can calculate. And let's say they are saying there is a question that in the food item, you have the carbohydrate, protein and fat. 10 gram carbohydrate, 20 gram proteins, 5 gram fat is there. So they can ask you the physiological value, they can ask you the calorific value. Sometimes they even add other things also, that your food is containing fiber, this and that, this and that. So that's your homework. You will solve 10 questions on the basis of this calorific value. You can Google it, you will get the questions for sure. Right? You guys are going to get the questions for sure. Clear? So next is the absorption. See, absorption, you know that we have holozoic nutrition, isn't it? We have holozoic nutrition, isn't it? Isn't it? So first step is ingestion. Second is digestion. Third is absorption. Fourth is assimilation. And fifth is egestion. Fifth is egestion. Right? So now the next step is the absorption. So absorption can be like simple diffusion can be there. Active transport where ATP is involved. It can be facilitated transport where the flow is as per it is, but the movement is down the concentration gradient, but the carrier proteins are involved. Clear? So absorption is the process by which end product of digestion they will pass through the intestinal mucosa, and maximum absorption is also there in the intestine. Right? See, if I talk about the maximum digestion, then it is in the duodenum. Your duodenum is having maximum digestion. But if I talk about the maximum absorption of food, right? If I talk about the maximum absorption of food, so that will be there in the jejunum. That will be there in the jejunum. Clear? Clear? Maximum absorption is there in the jejunum. Done? So you know that passive, active or facilitated transport can be there. Now, bache, passive. Passively. Passive means no ATP will be used. Right? It is along, or you can say that down the concentration gradient. When the absorption is along, or you can say that down the concentration gradient, then it is the passive transport. Then it is what? It is the passive transport. Like let's say here in the lumen. Let's say here in the lumen you have more glucose. Right? In the cell it is less. So from lumen to cell it is moving. So it is moving as per concentration gradient without the usage of energy, without the usage of ATP. So it is passive. So what is it? It is passive. Clear? Now, what about the active? A for active, A for ATP, A for against. A for active, A for ATP, A for against. I am repeating this again. A for active, A for ATP, A for against. Now here what is happening? Right, it is against the concentration gradient. Like let's say here in the cell, your glucose is too high, and here in the lumen, your glucose is less. But still, what am I doing? I am transporting this glucose from lumen to cell, from its low to high concentration. Right? I told you na, against the concentration gradient. So low to high it is moving. So I'm moving, doing something against the concentration gradient. It will take a lot of energy. Like let's say all are doing the same thing, but you have decided you are going to do something different, right? So obviously it is going to take a lot of energy. You have to convince yourself, you have to convince your parents, and you have to face many challenges. Same as the case here, right? So from low to high it will move, right? And here the movement will be with the help, it will be from the carrier proteins, right? Carrier proteins. The movement will be there from the carrier proteins. You know, na, plasma membrane is having some channel proteins, carrier proteins by which things will move. They come in and go down like this. And now when it is the facilitated transport, when it is the facilitated transport, when the word is facilitated transport, in that case, again, this is the passive movement. Again, it is down the concentration gradient. No ATP will be used. But the only difference is that here, bache, the channel proteins of plasma membrane will be used. The carrier proteins of plasma membrane will be used. Like my particular molecule cannot cross the plasma membrane directly. Here my particular molecule cannot cross the plasma membrane directly. It requires a tunnel protein. It requires a carrier protein. Okay, it requires a tunnel protein. It requires a carrier protein, right? So it's passive, down the concentration gradient, and no ATP is used. So these three words are written here, right? So like your glucose, right? In your notes, that's what you need to mention. Glucose, amino acid, and some electrolytes like chloride ions are absorbed by simple diffusion. So please in your notes include it that your glucose, amino acids, right? Glucose, amino acids, and some electrolytes, chloride ions by simple diffusion. Okay, right? But the passage of these substances depends upon the concentration gradient. Right now I am saying that your glucose, amino acid, they will follow that passive transport part. But it depends upon the concentration as well. Might be sometimes the concentration is less outside, more inside, but still I want them to come inside. So obviously in that case it will be active transport. So it depends upon the concentration gradient. So just see, glucose and amino acid, I told you, they are absorbed with the help of carrier proteins. So it is the facilitated transport. Like your glucose and amino acid cannot control your plasma membrane directly, cannot cross your plasma membrane directly. They need channel proteins. That is why the word is facilitated transport. But always remember, if it is facilitated transport, it is still passive. Okay, it's still passive because no energy is used. So transport of water depends upon osmotic gradient. It is going to follow the osmosis, movement of solvent from its higher concentration to lower concentration through a semi-permeable membrane. So it is clear. But some nutrients like amino acid, glucose, electrolytes, they are absorbed into the blood by this mechanism. Okay, so next, so see this line, that active transport occurs against the concentration gradient. So sometimes bache, amino acid monosaccharides like your glucose, they can also follow it. So it is not fixed that always glucose absorption is passive. It can be active. It depends upon the concentration gradient. Clear? So amino acid, glucose, they can move by simple diffusion as well. They can use ATP as well. It depends upon the concentration, right? It depends upon the concentration. And now when it comes to the fatty acid and glycerol, I have already explained, they are insoluble, cannot absorb in the blood. So they will first incorporated into small droplet called measles. They will move into intestinal mucosa. They will be reformed here, right? The fat globule will be covered with the protein coat. These are the chylomicrons. Then they will be transported into lacteals and finally in the blood, right? I told you, yes, Jyoti, we have already discussed this part, right? So in the mouth, stomach and small intestine, large intestine, everywhere there is the absorption. Maximum absorption is in the small intestine and particularly in your jejunum, right? Particularly in your jejunum. Clear, bache? Clear, bache? So obviously absorbed substance will be utilized by our cells, by our tissues for their activity. This is assimilation and the waste will be defecated. Okay? Okay? So digestive waste, you know that, stored in rectum, then a neural reflex will be initiated and there will be an urge or desire for its removal. So that's how they will be removed. Clear, bache? So peristaltic movement will help in there, right? So see the summary of the absorption in different parts. Even this table is important. So mouth may certain drugs which are coming in contact with mucosa, right? Or on the lower side of tongue, they can absorb into the blood capillaries directly. You might have seen that doctors sometimes they give the tablets which we have to keep here under the tongue and they will be directly absorbed, right? There is no need to ingest them. Mouth, right? So in the mouth, that is how they will get absorbed. And in stomach, your water, simple sugars and even alcohol. But it's a MCQ. Alcohol will be absorbed in the stomach. Clear? Then small intestine में, right, glucose, fructose, fatty acid, glycerol, they all will be absorbed. And in large intestine, water, some minerals and drugs. So question can come on the basis of this alcohol part. So alcohol absorption is there in the stomach. Clear? Alcohol absorption is there in the stomach. Done, bache? So now next is the disorders, the last topic, of course. So the inflammation of intestinal tract is the most common ailment due to the bacteria or viral infections, right? So you know that, right? If I talk about your ascaris, it used to live in our intestine, right? So there are different type of worms which used to live in our system and they destroyed. Like if I talk about the Taenia solium, obviously with the help of its hooks and suckers, it will even get attached to the small intestine. So obviously it will cause the problem, right? It is going to cause the problem. So we have many disorders. So first is jaundice. Question can come from this part, bache. So you know that here the liver is affected. Why? Because your villi, yes, villi, remember rubin, bilirubin will get accumulated in the blood, right? Body will become pale. So here liver is not functioning properly. When it is the vomiting, it is the ejection of stomach content through the mouth, and it is this reflex action is controlled by vomit center in medulla. Important question, right? And when you have just feeling, you are not vomiting, right? You are not puking, but you have just that feeling that yes, I want to vomit out. That is nausea. Okay, that feeling before vomiting is nausea, right? Right, it is nausea. Clear, bache? Then diarrhea, you know that watery stools, here the peristaltic movements are too fast. Clear? See, the abnormal frequency of bowel movement and increased liquidity. So here basically diarrhea, it's the watery stools. You know na, diarrhea can be caused by entamoeba histolytica as well. In biological classification, I told you, entamoeba histolytica, right? Amoebic dysentery it can cause. Okay, so here bache, peristaltic movements are too fast. Okay, in diarrhea, peristaltic movements are too fast. Frequently, watery stools will be passed out of the body. When it is constipation, right, feces are retained within the colon because the bowel movement here is irregular. So here the peristaltic movements are not proper. Clear, bache? They are not proper. And finally, indigestion, bache, when food is not properly digested, right, the enzyme secretion is not proper, or there is the overeating and the spicy food. So I really don't think that we need to explain it in detail, right? So these things are the basics. Hana bache, these things are the basics. Hope you know about it. Fine. So that's how the absorption will be there, and this is all about the disorders here. Okay. Okay. So next and the most important is PEM, protein energy malnutrition. That's important, bache. It's PEM, protein energy malnutrition. Here we will talk about the marasmus and we will talk about the kwashiorkor. Right? Do not miss this part. This is very important. So here we have the marasmus and the kwashiorkor. But a dietary deficiency. When in our diet, proteins and total food calories are not proper. When in our diet, we are not taking proper proteins, if we are not taking proper calories, so it can result in protein energy malnutrition. Right? So in underdeveloped countries, just say you can say that South and Southeast Asia, South America, West and Central Africa, people are having these dietary deficiencies because they are not getting the nutrient enriched food. So it can result in these protein energy malnutritions. Are you getting it? Are you getting it? So here, right here, like see, they have given the examples here that PEM may affect large section in the population during drought. When there is the drought, when there is the political turmoil, issues like that, right? Let's say for a long time the rain is not there, so crops will not be properly harvested. So obviously it can also result in the deficiency of these essential food items. This happened in Bangladesh. They have given the example of Bangladesh during the liberation war and in Ethiopia during the severe drought in the mid 80s. So they are just giving the examples here, but ultimately what you need to focus, what should be there in your notes, marasmus and kwashiorkor, right? So these two disorders, they are because of this deficiency of these proteins and the calories, protein energy malnutrition. जैसे for example marasmus, deficiency of protein is there बच्चे and calories. मतलब simultaneously both the things are absent, proteins and calories both are deficient here. Clear? So it is found in the infants less than a year in age, right? So if mother's milk is replaced too early by other foods, right, then this can happen. So in marasmus both are absent, proteins as well as calories. So less than 1 year of age, right? Infants less than a year in age, they will get affected with it. Clear bache? Clear bache? And this often happens if the mother has second pregnancy or childbirth when the older infant is still too young. That is why it is advised that there should be a gap of at least minimum 3 to 4 years in two children. Okay, so next is in marasmus, protein deficiency impairs growth and replacement. Of course, proteins are not there. Extreme emaciation of the body, like body will be very thin, right? There will be the thinning of limbs, the skin will become dry, thick and wrinkled. So here what you need to focus, the symptoms. That's very important to remember. So you should know about that comparison. So here you guys can see. So we were talking about the marasmus. So here you guys can see occurs in children who have an overall deficiency in energy intake, which includes even proteins. So proteins and energy both are absent, right? Muscle wasting, like muscles are very thin, right? Fat is not there in body because neither calories nor proteins are present in the diet. So here dehydration, total muscle loss, anemia, dry skin, brittle hairs, children are more irritable, and it will occur before 1 year of age. The infant which is less than 1 year in age. And in kwashiorkor bache, calorie intake is normal. See, K for kwashiorkor, calorie. It seems na kwashiorkor calorie. So calorie intake is normal, but protein deficiency is there, right? So because of that, here the best thing that you need to remember is edema. But what is the meaning of edema? Edema means swelling, right? So here calories are proper, proteins are not proper. So in kwashiorkor you will see edema will be there due to water retention and distended abdomen. Like there will be the swelling here. Abdomen is distended and the swelling will be there. These two things, right? You need to focus on these points. Then only you can differentiate which one is kwashiorkor, which one is marasmus. So in NEET paper, they can give you, right? In the NEET paper, they can give you the symptoms and they can ask you which disorder it is. Clear? Thank you, Bache Sudama. Okay? So growth rate and body weight decline in the marasmus. Okay, even the growth of and development of brain and mental faculties are also impaired because neither energy nor protein they are getting in their diet. When it is the kwashiorkor, right? K for kwashiorkor, calories are there, but protein deficiency is there. Okay, so let's say mother's milk is replaced with high calorie and low protein diet. That is why it is happening. So child more than 1 year in age will get affected. So marasmus less than 1 year, kwashiorkor more than 1 year. Clear bache? So you know that there will be again the wasting of muscles, thinning of limbs, failure of growth and brain development. But here what will you see? You will see that some fat is there under the skin, right? Edema means swelling. Swelling of body parts will be there. So if swelling, kwashiorkor, no swelling, totally thinning, that will be marasmus. That's all. Clear? That's all. So this is all about this chapter. Right, so finally we are done with this chapter. So here you guys can see the tables and see the hormones are also there. So gastrin, stomach stimulate gastric secretion. Enterogastron, also known as gastric inhibitory hormone, released by duodenum, acts on stomach, inhibit gastric secretion. Secretin, first hormone discovered, released by cells of duodenum, acts on pancreas, liver and stomach. So stimulate bicarbonate secretion in pancreatic juices, increase secretion of bile. Then cholecystokinin, pancreozymin, right? So it stimulates the contraction of gallbladder and pancreas. But this is a theoretical part that you need to remember. Okay, this is a theoretical part that you need to remember. So I'll share the PDF in the Telegram group, right? You can join my personal Telegram group, that is Love Biology by Ambika. So I will share the PDF group, PDF there. Okay, but yes, try to make your own notes as well. Okay, so yes, as promised, we are done with this first chapter of human physiology, that is your digestion and absorption. So Sunday at 1 p.m. we are going to practice top 50 questions of digestion and absorption chapter. Right, next week breathing and exchange will be done, and yes, Monday you will have your YouTube class at 10 a.m. in the morning, and that will be on sexual reproduction and flowering plants. Okay, done. So we'll share the PDF as I said. So what you have to do, if you are the part of that Telegram group, fine, otherwise join it, and please now revise this chapter from NCERT and solve at least 100 questions. Okay, so tomorrow what you guys can do, you can revise this chapter, and Sunday we will have a practice session at 1 p.m. Okay, so within 1 hour we will solve all the questions. Okay, within 1 hour we will finish it. So top 50 questions of digestion and absorption will be practiced. So thank you so much for your time. Now you know what you have to do. In the comment section, I just want one comment that you like the session or not. That's all. So you know that it is not going to take much time. So do it quickly right after the session. So take